And I want to ask everyone, if you have questions during the presentations, to please put them into the chat window. And we will have people who will be answering questions as we go along in the chat. And also the presenters will can answer questions from the chat as well. Also, before the end of the workshop, you will receive an email with a link to a SurveyMonkey evaluation of the workshop. We would really, 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 really appreciate it if you would fill those out. It's very valuable information to us to have in helping us to continue to always improve our workshops. So without any further ado, I'm going to turn things over to Mike H., which is our first presenter for the day.
Thank you, Michelle, and it's my great privilege to be able to participate in the NEMO workshop for the second year and to give this introduction. This is a great resource, a great community that is serving, amongst other aspects of the BRAIN Initiative, the BRAIN Initiative Cell Census Network, and these workshops are basically centered around looking at this data, working with this data, and using the NEMO resources. It's structured as a two-day event, two half-day event, largely for convenience for people, and so as to avoid intensity and kind of in the kind of presentations. And the first day today, this is today, in addition to, I'm just going to sort of give a brief introduction to the workshop and to some of the data sets involved in the workshop. And then most of the rest of the session will focus around learning about the tools and resources for looking at the data, visualization, and analysis, including NEMO Analytics, EpiViz, and various sort of other kind of in essence, third-party tools, if you would, which are basically typically things which had been developed from people in the consortium or our partners. The second, coming up next Thursday, essentially a week from today, yesterday, this is more, will be the more, today's workshop doesn't really require, in particular, computational expertise. It's meant for the scientific user. Next week's is a little bit of a more kind of data intensive kind of workshop where people will learn about the data processing tools. It will include a deep dive using kind of these data sets in Terra, the NEMO portal, and running your data, running workflows, and I know that Tim Tickle from the Broad will have quite a bit to say during that workshop. So, there's a lot. The NEMO resource is the primary resource in Brain Initiative data for omics now, and contains an immense amount of data from our partners in the BICCN. You will learn about how to get this data. You will learn about how to interact with this data. So I won't go into too much of that now. This is part of what we'll hear. I'd like to emphasize that some of the NEMO family is far more than just data access and archiving, but includes many kind of tools and methods for scientifically examining the data, looking at the data, and working with this data. In particular, the NEMO Analytics, which is a great facility for studying and visualizing relationships amongst the primary data sets. So I know you'll be learning about that today, from Rona Herzano and Brian Erb, and I hope that you take advantage of that. Just a citation of the paper, basically this resource, if you look at this paper, came out in Nature Methods this year, and it will give you a nice overview of their capabilities. The NEMO archive is also part of this community ecosystem of the BICCN that we have, the Brain Cell Data Center, which I work with at the Allen Institute, helps to kind of organize and kind of encourage the different participants to kind of practice sort of open kind of science. So if you go to BICCN.org, you can get a feel for all the different investigators who are part of this now. We tend to have kind of basically data kind of grant pages for them, which give the basic structure of their data sets and their awards. This is a illustration of from the BICCN the major data sets in NEMO, and this includes basically mouse, human, marmoset, omics data, including transcriptomics, single cell data, single nucleus data, epigenetic data from the adult and developing organisms. This just sort of summarizes the principal These were all kind of major initiatives as part of the BICCN, which is coming up to kind of in its final year. We'll learn about how to interact with some of these data sets in this workshop. So just a little bit of a survey of kind of what was done and where we are with this. So on next Thursday as well, October 7th, although actually October 6th it appears online, this major package of the results of all this work of the consortium, this sort of first kind of highly collaborative and highly a really deep dive into the multimodal analysis of cell types in the mammalian primary motor cortex. It will occupy an entire issue of Nature. The flagship paper is called the Multimodal Cell Census, an atlas of the mammalian primary cortex, and contains hundreds of authors who did all the work of this wonderful study. Picture here shows a figure from one of the extended data figures showing the overview, which contains all basically the experimental and the computational approaches that went into this analysis. And I'll talk about some of these as we go through. But this is kind of these are the kinds. A lot of the omics data sets are the ones that we'll be working with here today. The resources associated with that are kind of orchestrated through the Brain Cell Data Center, and it includes the NEMO archive as a primary and main kind of archive associated with this work. There's also, on the imaging side, the Brain Imaging Library, housing microscopy and imaging data, and the DANDY, our neurophysiological archive, which contains cross-modality data, patch-seq data, and other kind of functional imaging related data sets. We organize these at a project and specimen level at the Brain Cell Data Center, and try to direct people toward the various tools, including common coordinate frameworks, browsing and visualization capabilities, and other things. We will be releasing also. I won't show you here. It's kind of still under a minor embargo, but we will be releasing a kind of a splash organization site associated with the publication, which gives access to all the papers. Will point to tools that are sort of related to interacting and managing with this data, and it's sort of a one-stop kind of place for understanding the work of the consortium in this effort. Just a few, let me make a few, I don't want to take away from the interest in digging in deeply into the data sets, but I'd like to just take a few minutes to talk about some of the nature of some of the findings and the results. So at the heart of so basically the major work centers around looking at one area, the primary motor cortex. Because of its key homology and essentially ready identification across mammalian species. Other areas like the visual cortex will have more kind of potentially variable locations and sophistication in different organisms, but the motor cortex is a fairly stereotyped region that made a nice kind of substrate for really examining a mammalian brain kind of cell type plan. a key part of this is a transcriptomic and epigenetic kind of analysis of primary cortex, motor cortex, and it includes a cross-species and deep dive of both using single nucleus, single cell RNA-seq, as well as the epigenetic data. And there's kind of really good alignment found between these modalities, and it's sort of a rigorous analysis of the types. The results can be presented in a kind of a taxonomic organization where one has various confidence levels of understanding about what level of the tree one can be understand the kind of statistical significance of, and essentially the number of clusters found by species in human, marmoset, and mouse. And the interactions between those species will play a key part throughout the paper. So basically, and there's also the identification of defining marker genes and express genes related to differentiating these clusters in a stream. All this data in particular can be viewed in the NEMO analytics, and I know that the NEMO analytics built out a very nice kind of tools dedicated to each of these data sets, which you'll be learning about later in our workshop. So this paper, let me just one more comment, is that the Nature package consists of the flagship paper that I mentioned, as well as 17 kind of core companion papers. Actually, a couple of them appear earlier in Nature, but they're considered part of that, and they will appear on the BICCN splash page as core companion papers. But this paper in particular, transcriptomic and epigenetic analysis of mouse primary motor cortex, Yao, Liu, Xi, Fisher, through organized and kind of overseen by Honghui Zheng and Eran Mukammel, is one of these papers. Another key approach, which is in increasing and in our ability to do it in a rigorous and high throughput manner, is the MERFISH technique, which now is actually even available as a commercial platform, and is proving to be highly kind of successful in understanding the spatial distribution of cell types. So here, this was used in this work in two ways. One was the analysis of cell types was done independently, so that one derived the actual cluster and taxonomic relationships using this modality alone. And then they were compared and mapped together to understand the relationship and affirmation kind of with the other kind of the primary kind of RNA-seq. The nice thing about there's again applications of this are available through the NEMO resources, but the idea here is that one can understand in deep ways sort of cellular kind of architecture in the brain and location, and it's a common understanding that as this method is scaled up in the next series of awards that we'll be doing in human and primate, that will be a key determinant of understanding actually the cell types of the human brain. One more comment, sorry, that will appear as the paper Spatially Resolved Cell Atlas of the Primary Motor Cortex in the same package. So in connecting, a nice way to understand the kind of functional kind of implications of cell types is to study morphoelectric correlations with these data. In other words, what cell types, what do the morphological and physiological patterns have to say about cell types, and how do they relate? Whereas this is a complex landscape, a great deal of kind of coherence and association at the subclass level was found in the study of Andreas Tolius and their lab. And this paper, again, will appear as part of the main companion papers. And this is what I'm showing here is actually several figures from this flagship paper, which went into the wrap-up and interpretation of this data. We won't be talking much about patch-seq data today, but it is important to understand this as a important kind of part of the big three, if you will, or three or four of cell type identity. And certainly another important thing is to understanding the epigenetic landscape of these data. Here, in two different kind of studies, both which appear as companion papers, there's an atlas of which is also called the SEMBA atlas, that SEMBA atlas, which comes out of the Ecker lab, is a study of DNA methylation in the mouse brain at single cell resolution. And these are data in NEMO. I know we'll be hearing about some of this data today. And also use of this in combining it with kind of essentially retrograde tracing to understand projection targets, and at the subclass level, what connects to what and with what, essentially with what frequency across the brain. This was a key part in resolving kind of connectivity and its relationship to cell type. So these all, this fits together in a kind of major sort of wrap-up analysis that shows for the final figure in the flagship paper of a kind of common taxonomy across species, together with understanding its variability, with its association and determining of morphological types, and the ability to kind of understand the variability across these different platforms and these different methods. So we believe this to be, a definitive plan of cell type organization in a key area of the brain, and one for which I think it's fairly commonly understood that this is not just a single area, but performs an important blueprint and a likely kind of foundation and basis for the actual cell type plan of the mammalian brain, which we hope to explore more in upcoming awards. So let me just wrap up by welcoming you to the workshop and thanking our NEMO team, which has done a great job. It's been my privilege to be associated with this group, and, especially Owen White, principal investigator, Seth Ammon, Michelle Giglio, Anoop Markohar, Lynn Shrimi, Heather Creasy, who I interact with a lot via the BCDC, and all our other staff here. So thank you, and I hope that you enjoy our day.
Thank you very much, Mike. Any questions for Mike? All right. We will go ahead and move on to our next session, which is on NEMO Analytics and EpiViz. Presenting in this session will be Brian Herb and Jairam Kancherla. So I'll let you take it away, Brian.
All right, thank you very much. So throughout this session, we're going to be bouncing back and forth between Jairam and I. I'm here at the University of Maryland, trying to do mostly the genomics side of this, and Jairam is a member out at Genentech, and he is still working on the EpiViz platform, which at its heart has an epigenetic browser, but it's a lot more than that. There's a lot of good analytical tools that go along with that he'll describe. So if you have not seen it yet, in the chat, I've listed two URLs, one being this link to the Google Doc here. And that's our first link. And we'll simply be working through this schedule for today. And then the second one is the actual site, NEMO Analytics. And we'll first go through just a couple of intro slides, and then we'll show you how to log into NEMO Analytics to explore the profiles. So as we work through this, as Michelle noted, if you have any questions, just place them in the chat, and somebody from the NEMO team will answer. And as Mike mentioned, it's an absolutely impressive amount of data that the BICCN has been producing over the last 5 or more years. NEMO Archive is hosting all of the genomics data. So all this at the top, the single cell epigenetic and transcriptomic data. And with the real goal of trying to understand all the cell types across the entire brain. And the data that's in the archive right now, there's pilot data on almost everything across the human and mouse brain, which is very, very impressive. And it's a lot of that is public right now. I encourage you, if you do have your own project, explore that space, see if there's other data sets that may be used in conjunction, and really start diving into that data. Other archive sites, Dandy, Bill, they're hosting most of the other electrophysiological and imaging data. And hopefully within this BICCN ecosystem, we're really hoping that users have the seamless experience of bouncing between all of the data and all of the tools associated with them. And as noted, there's a whole host of papers that are coming out this year. Again, a lot of them focusing on the pilot project of looking at the motor cortex. There's a very nice paper on cross-species comparison, and also simply taking a deep dive into the transcriptomic and epigenetic data within the mouse, within the motor cortex. And starting to be a couple papers on this whole idea of comparison across brain regions. So what we're trying to do at Nemo Analytics is both support these publications, and the idea would be if somebody was coming to Nemo Analytics from the paper, so they read the paper, they see these beautiful t-SNE and U maps, and there's an understanding of what the cell types are, perhaps there's marker genes, and from that point, if you're trying to integrate this in with your own study, what Nemo Analytics tries to do is take those conclusions from the paper. You can take that UMAP directly and hopefully merge in your own data set and compare it to these established published data sets. And again, just kind of an overview of the Nemo architectures here. So we have the archive itself. You can go through and select files based on a faceted search. So you can select it based on study, species, brain region, data modality, anything that we have available metadata for. And if you're coming to this from a certain publication, we also have an organized landing page for every single publication. So this is an example from the mini atlas paper. There were 9 data sets that were identified in that paper, and they're organized by raw data. So you don't have to wade through the massive terabytes of all the data associated with that paper. You can go in and just look at perhaps the ATAC data or the single SmartSeq data. And so we try and have this organized so that you can really get to that data set and also actually cite that data set individually. Each one of these has a Nemo identifier. The other nice thing is that we try and organize the analysis files as well. And this is kind of something that genomics up to this point has been a wild west of, what kind of data has to be submitted per paper. A lot of times it would be the raw data to SRA, but we're also trying to wrangle all that analysis data where if the paper concludes on saying like this is a certain cell type, or this is the clustering map that we have for these cells, we're trying to capture that in the analysis files as well. And that's super useful when you go and try and use that data for a new study. So all of this data is, we're curating it and drawing it into Nemo Analytics. And the idea is that within analytics, you have all of these data sets available to you for comparison. And we're heavily emphasizing visualization tools. So whereas you may have just a single view of a single data set, one thing that you can do in analytics is stack multiple data sets next to each other, make a comparison based on the gene level, and you can even compare between transcriptomic and epigenetic data as well, as we'll see in a couple of these profiles. We also have some tools. So if you had a new single cell data set, you can go through and do an analysis on that, clustering, find the marker genes, and then again, compare it to these published data sets. So that's going to be the heart of this first portion of the workshop, is how to go about navigating Nemo Analytics. So a couple examples of these profiles that we have created for supporting the papers. Again, this is just looking at the motor cortex mouse study. This is an example of a mixed data. So we have the transcriptomic data up at the top. This is their, in this case, just the 10xV3 samples from the Allen Institute. And when you type in a gene, what you see is where that gene is highlighted in this established t-SNE. And again, this is using that clustering exactly from the paper. and their cell type designation. So very quickly you can see where your gene of interest is expressed and what cell types are expressing it. But as Jayram will introduce to you, we also have EpiVis panels for these guys. So that exact same cell clustering. So what they were able to do in this paper is actually integrate all of this into the same space. So you have the same identified cell type in the single cell and on the transcriptomic as you do the epigenetic. So you can very quickly see what are those cell types that have open chromatin regions in the promoter region of that gene. So once you type in that gene, you actually get directed in the genome view directly to that gene. And you can see very quickly open chromatin regions, and they also have associated DNA methylation. So you can see if there's a associated demethylation events over that gene, which all leads to increased gene expression. Here's another example of mixtures of data types. So for the cross-species paper, they also did ATAC sequencing across those various species. You can compare them directly. The other nice thing about neoanalytics is that you have a lot of leverage over the types of plots that you can produce. So this top example is a plot that was not presented in the paper, but using their data, we've made a bar plot of per cell type, what is that species comparison? So for our given gene, I think this is SCAD2 yet again, you could very quickly see, is there a species difference in gene expression within a particular cell type? And so you can produce bar graphs, you can produce violin plots. Essentially, once you have that raw data, you can produce a whole host of graphs that they can place next to each other in this profile. Since there's a great interest in spatial data, we've also introduced a few profiles using the MERFISH data this is now from the flagship paper. And as you see right here, this is the cross-section of that motor cortex region. They have identified a couple of individual cell types. You see them beautifully stratified throughout the cortex. And using their MERFISH data, this is just simply a plotting of that data. So you can quickly see that there's an enrichment of, in this case, TET-M3 in the L2, L3 layers. And to make this a little bit simpler, again, this is just another type of plot that you can produce. You can take this data, summarize it into a violin plot, and very quickly confirm, yes, indeed, layer 2, 3, and even dissecting that further, you see that's only in these two subtypes. And then lastly, a very specialized type of data that we can add in is you can actually use an SVG image and overlay data on top of it. So coming from the patch-seq data, which takes both the morphology, electrophysiological signatures, and then it actually pulls out that nucleus into single cell transcriptomics on it. For all those different cell types, you have that morphology, and then you can also have a summarized gene expression. So again, typing in GAD2 here, we can see which cell types are expressing GAD2 the strongest, and you can see that associated patch-seq data as well. And we really hope that the Analytics platform is a good choice for folks that are publishing, to have this nice companion where people can explore. So an example coming from the cross species, what they gave us were those count files, cell type assignments, the UMAP coordinates, and any the colors coming directly from the paper. And all of that information can simply get created into a profile. And we see that those exact coordinates, those exact colors are now presented. So the hope is that it's a seamless experience from the user reading the paper, going to the site, and then they can really dive into the data themselves. And as I mentioned before, there's other plotting options that are available. How this can work in the paper itself, and this is something that we have integrated into the flagship paper. At the bottom of each figure, you can actually put in that short URL, and that goes directly to the specific profile that you want. And a couple of quick tools that we'll go over a little bit later. We have simple comparison tools. So if you have within a single data set different cell types, you can do just your typical scatter plot, look for the most differentially expressed genes. For single cell data, we have the entire workbench, which is based on the ScanPy pipeline. And we're going to walk through this together after the EpiViz presentation. But essentially what happens here is, again, you do the same QC steps that you might do in a Srot pipeline. And you move through and you can do your clustering assignments. Once you have the clusters, you can identify top expressed genes. And by simply selecting those top expressed genes per cluster, you can produce dot plots, violin plots, or if you wanted to do that same comparison of this cell type versus that cell type, you also get some nice violin plots for that as well. And you can export all of this data. Okay, so just a quick thank you to all those involved in this. All right. So at this point, we're going to move into a more interactive part of this presentation. So if you don't mind, follow along with the schedule. And I'll just be going through and clicking as the participants are clicking as well. And so we're going to first go to the NEMO Analytics site itself. And if you have not done so yet, click on the Create Account in the upper right-hand corner. All it does is require an email. As Joshua Orvis has mentioned in past workshops, he only emails maybe like once a year. So this is definitely not going on any list. It's just for the purpose more of getting you an account here. And so as folks are creating the accounts, I'm just going to go over a few of the features of the front page. If you ever need to get back to this home page, you can always click on the NEMO in the upper left. It is, the site itself is very gene focused. So if you click on this upper left-hand box here, type in a gene symbol, it does a few things for you. One is that if you simply typed in, say, like SOX, and you would get that entire gene family. But if there's a specific, you only want SOX2, type in SOX2, hit the exact match, and your search then would only look for that single gene. One thing that a new feature that will be coming up in just perhaps probably over the next month is the ability to hold and search upon multiple genes. So some of that functionality is already established. You're going to see a lot more multi-gene functionality in the future. Right below this, we have the different profiles. I think we're going to be going over what's the motor cortex? Motor cortex cross species is one of the first things we're going to tackle. And so I'm going to click on motor cortex cross species, and that'll select that profile. And then once I type in a gene, I will go directly to that profile itself. A few other quick items as we're on the front page right here. These will be the tools and utilities we'll dive into in just a little bit. There's that single cell workbench that I mentioned. If you need to have any help with the manual, there's a whole host of videos that we've already walked through all the different aspects of this. Gear, by the way, is the underlying engine of this website. So there was a nice recent Nature publication on just the actual platform itself. And this is a very adaptable platform. You can use this in any other types of data that you want. And again, more manuals there. And there's that recent publication. All right, so if you have any questions or you need me to slow down, please write those in the chat. Other NEMO folks will help you out. I'm going to go to that motor cortex cross species. I'll type in GAD2 and I'll click on exact match and search, and that'll jump right to that profile. Okay, so we're now here in the schedule. Okay, and since everyone's jumping on it, maybe a little slow, but you see those exact same pictures I showed you in the slides. So this type of display is that, well, we call it the static T-stain display. In this case, you can add on a figure legend right next to that dynamic T-stain showing the expression of the gene. And so for the cross species, we have a human profile, a mouse profile, and a marmoset. And again, as I was showing you before, we have the comparison. This is organized by the individual neuron subtype. And you can very quickly see if there's an overexpression of a given gene for a single species. So something's going on with the layer 6B cells in mouse that has a higher expression of GAD2 than the other species. So that's kind of nice to see at a very quick. For every single panel here, we have a couple of items on the right-hand side. So again, just as a quick overview for the page, you have some annotation at the top. So you have annotation by organism. Based on the data set, it already knew that this was a mouse data set that's within the metadata. I'm sorry, well, it's within here. So if you needed more specific information for GAD2 and human, you simply click on the human portion and you get a little bit of that gene information. And there's also link outs to various other either data resources or other gene information. So the panel itself, the one thing is this little pen tool right here. If you wanted to take a note on a specific data set, you can, and that'll be saved underneath your profile. So if you were to work with this data set a little bit more and say like an interesting note about, oh, well, GAD2 is expressed in these LAMP5 cells, and you wanted to make a special note of that, that's a good place for putting that. In this drop-down menu right here, there's other things that you can do with this particular data set. So if you go to displays, there may have already been other plots that are based on this particular data set. So it looks like other folks have made other type of bar plots. There is an old clustering or sub-clustering. So right now, this is the default, but you can actually have other plots made of the same data set. This will be simply info on that data set itself. So actually pulling a lot of that from the literature. This describes how the experiment was done, what types of cell types were found within the data set. Information on the publication, which should be updated very soon. So if there is a, if that is already in PubMed, which I don't believe it is just yet, that may be linked right there. The geo entry, if that does exist. The compare tool is that scatter plot that I was showing you before. So if you just wanted to compare like astrocytes and endocytes, that would be a very quick way of getting to the DEGs. The curate is something that we'll do a little bit later on, but that's where you can go in and make new plots. And then analyze takes you back actually to the single cell workbench, which we'll be going over in just a little bit. So hopefully there's a lot of, oops, sorry. So that's something we'll get into a little bit later with the single cell workbench. So a couple of options. I mean, even if you're right there at this view, you can take a quick dive into that particular data set.
Brian, we have a question.
Yes, Sean. Mm-hmm.
Someone was asking, when you look at differential expression in the gene for different species, they want to know what the y-axis is specifically.
Yeah, I think that could be altered based on how you're presenting the data sets. I think this is just purely, this may be at this level, the counts. Now, I think if you wanted to, you can put in a normalized counts for this guy. So
yeah, this is raw expression, but if you have a column in your data set to normalize, you can certainly use that, elect to use that instead of the raw expression.
Right. So the proportion may be correct. So that is one nice thing about when uploading data. You can have a raw data set, you can have a normalized data set, and then when you're creating these plots, you can select any one of those. So it's fine. Okay. So we've done that. We're going to take and if any other questions, so that was a more straightforward UMAP view, very akin to what you would see with typical single cell data. We're going to take a quick jaunt onto the patch-seq profile and just take another look at this because it was a little bit cut off in the presentation. But again, this is something that you can have a lot of latitude with. The SVG image, how I built this profile, I simply took from, in fact, the companion paper, this view of the data. So this was just simply in listed as a large PDF. I took those various elements from them, and then within the SVG itself, built these boxes, and then assigned the values to the boxes of the transcriptomic data. So in this way, you can imagine there's a lot of options where you can have maybe a display of a neuron where you're showing various aspects of it lit up. You can have an image of the brain, and you can have expression across the brain. So almost anything you can put into a PDF, that itself can be layered on top of it, gene expression information. So again, that's just simply that average expression across that data. And then for the MERFISH data, that is something that came to us as it had XY coordinates for every single cell. And coupling that with the cell type assignments, this is just simply actually a scatter plot. And then you can assign to the dots, either in this case, categorical information of the cell type. You can link to it the gene expression information. This is what you saw in the presentation, a little bit granular breakdown of those neurons. And again, you can simply create a violin plot out of that. So I think one that expresses a little bit better here might be CUX2. I'll search for that. Notice that here is a place where I can toggle that exact match on or off. Yeah, and this one shows up a little bit better in that layer 2, 3. But MERFISH data in particular, there's about 250 genes that they interrogated for that particular study. So if you go through the MERFISH data and you type in a gene and it's not found, that's most likely why. It's just simply not something that was interrogated in the MERFISH technique itself. Okay, so that kind of goes over the basic profiles of NEMO analytics. We're going to pass off to Jairam for the section on EpiViz, and then we'll come back with the single cell workbench.
All right, I'm sharing my screen. Can you see my screen?
Yes.
Yes.
Okay, I can't see, sorry. All right, can you still see my screen when I present it or no?
You're good, yes.
Okay. All right, so Brian provided a good overview of everything that NEMO Analytics does. I think the next session of the workshop also talks about how you can process the data through the Terra platform that Tim is going to handle. But it shows various features that NEMO Analytics provides, where you can look at from the single cell transcriptomic data, the expression of different cells and their clustering, but also look at their epigenetic data together if you have the same kind of cluster mappings. All right, so coming to EpiViz. EpiViz is similar to a genome browser. If you're not familiar with genome browsers, they use the they take advantage of the linear organization of data across the screen, and they visualize data as tracks. They also provide annotation information of showing you the position of genes, their promoter origins, or different annotations that are available from different consortiums or experiments. And they provide ways for quickly navigating along the genome. This could be, if you know what the region you're interested in, you could quickly type in that region in the application in the browser, and it would update all the data to that region, or you could search for a gene and quickly jump to that region. So APVS is also works very similar to a genome browser. We support a lot of both plot and track-based visualizations. And what I mean by that is, if you have expression data set or a transcriptomic data set, it is much more intuitive to look at those data sets as a heat map or a scatter plot, where you're comparing expression across samples and not necessarily as a track. So we support those kinds of visualizations. On APVS, all the data across different experiments is linked by the genomic position. So if you hover over one track, this could be on a heat map as shown in this GIF as an example, it will immediately highlight that region across all the other tracks. So this provides you an immediate visual cue of what that region looks like across data sets. And some of these workspaces that you create, or these profiles that you create as part of the NEMO Analytics platform, these can be shared within NEMO itself, and you can add them to various profiles. And Brian will show you how you can add these profiles to, how you can add the epigenetic panels to various profiles at the end of the presentation. We made a couple of workspaces for the data that is coming out of NEMO or BICCN, the consortium itself. But you're also free to upload your own epigenetic data to the NEMO Analytics platform and then create these visualizations. One of the features that APVS provides is, for example, let's say you have epigenetic data and you have clusterings generated at different resolutions. Our system can automatically generate a mean score across these clusters at a higher level, but can also visualize the data at the lower resolution. So instead of generating these epigenetic files at different resolutions, you can generate it at the lowest resolution possible, and then ask the system to do these aggregations. And these are some of these that we've done for the flagship paper. For the part of the workshop, I'll show you the same plot that Brian was showing before. How do I take this off here? So if you have access to the slides and click on this link, this should take you to the mouse MOP epigenetic profile. If not, I'll put this link on the chat. And once you're here, and if you're logged, this is a public data set, so you don't have to log in. You can search for your gene of interest, and we'll choose GAT2 for this use case. You can either do an exact match, similar to how Brian was showing in one of his earlier workspaces. And once the data comes back, you'll see that same profile where you can see the GAT2 expression across different clusters and which cell types they are expressed in. And in addition to that, you'll also see the epigenetic panel here, which shows you the center of this region is always the gene that you've searched for. So that's the GAT2 region. We add a little bit of window both in the downstream and the upstream of this gene region to see what other genes are in this are around this gene. And then you see the expression profile. The first is the ATAC-seq data, so chromatin accessibility across these different cell types, and the methylation profile across these cell types. So one thing APVS does is it renders the data as it's coming from a file. You can also choose to highlight. You can also choose to set the max value for each of these tracks here, which is around 1.5, and then you can clearly see what these labels are and what those different profiles look like. There's a few things you can do is, let's say for your interest, you're not interested in all of these different cell types, you can remove these, and that should hide some of these tracks away from the analysis. I'll also quickly walk through some of these. And so most visualizations on Epibis also have settings that lets you control what titles you want to put, what margins you have, but also mostly control the settings. So if the data is too dense in this region, or there's a lot of data points in this region, you can use the step parameter to scale down the amount of data that is being rendered. So it shows a step of 10, so that reduces the amount of it uses that as a smoothing function and then uses lesser number of data points to render. You can also decide to choose if you want to show the lines, if you want to show the actual data points itself rather than the smoothing parameters, and also control what kind of a smoothing function you want to use. We use some of these, we provide a lot of these commonly used smoothing functions like the linear, the basis is what we use by default as the smoothing function, but you have the option to choose any type of smoothing function. So that's the gist of what Epibis provides as part of NEMO Analytics. I quickly went through the interface, but a quick overview is you can also, there's a button on the top that lets you open just the epigenetic data in its own window. And then you have full control on many of the features that Epibis provides. Where you can quickly jump to a gene region, you can search for a gene, go to that gene region, or you can navigate along the genome, you can zoom in and zoom out, and also add your own custom tracks rather than the tracks that were added on this particular profile. Apart from what I've shared, I'll quickly go through some of these. So there's a notion of fixed axes versus not fixed axes. So when you get data when you run these experiments and get different ADAC data, most of these data have their own maximum value within the data set. So you can choose, when you create a visualization, you can choose that all the tracks choose of the same fixed axis, the maximum Y value, so that it's more relatively comparable across data sets. But if that's not the case, you can also choose them to not have a fixed Y value. So you're looking at a more data that is per data set or per track. You can also perform transformations. This is possible on our line tracks, where if you have, this is mostly useful if you have replicates or even a compared two different types of data sets. You can do a log2 ratio directly on the tracks. You can mirror the tracks one on top of each other, so you can see where the highest expression is or which data set contains the higher expression, or you can also calculate mean across all of these. And this is mostly useful when you have multiple replicates and you just want to see the mean of the data and not necessarily all the replicates. If this is some of the things I've shown before, where if you have data that is too dense, or you're looking at data at a very high level, you can use the step parameter to calculate an average for every 10 base pairs if the value is 10 in the window, and then visualize those rather than looking at the data at the base pair level. On heat maps, we also provide a lot of clustering metrics. By default, our the heat maps cluster by their by the sample or by the cell cluster. But you can also choose to either cluster by samples, by rows, and there are different methods that you can use there are different clustering methods that you can use to customize these visualizations. This is something I've shown in some of these plots. Not all of the plots have this. When you have too many data sets that you're showing, you have the ability to hide or show some of these tracks as needed, because maybe sometimes there's a profile that is already created in NEMO that contains like 20 different cell types, but there's only a few that you're interested in. So you can, instead of recreating that entire profile, you can hide some of these tracks that you don't want to see. You can also totally choose the colors that you want to use for your visualizations, and these are persistent. And so when you create a epigenetic panel and you update these colors or any of these settings that I've shown so far, those are always persistent, and that's how they get stored into the NEMO database. And when you come back and look at the data set itself or that profile, it'll always be the same state as where you left it out at. So to get your data into into the NEMO platform, there's two ways. One, you can go to NEMO archive, which hosts a lot of the consortium data sets and get the files that you want or the files that you want to explore. Or we also provide an interface that lets you upload your custom data sets or data sets that are available on your laptop or through your lab. And you can upload them to the NEMO Analytics interface. I'll show you where that is.
Fair, there was a question in the chat about how the names of traces that you can hide correspond to what was plotted.
How
I think that was a few slides ago.
Those log numbers and letter names to the beautiful. Which are these the ones? So yeah, so this is how the data is stored in NEMO archive. So the lab, at least when I first registered, that this is how it starts. It's the lab and the experiment. So this is an ATAC-seq experiment. When they perform the clustering, this is a major cluster, and then the cell type, and then I guess the major cluster again. Brian can correct me wrong if I said something wrong here.
No, you're fine.
Yep. All right. Was there anything else I heard? Okay. So to upload your own custom epigenetic data set, once you log in, and this is something that is only available if you log in, there is from the drop-down menu here, there is an upload data set, and you can upload your epigenetic data set here. So if you're trying to import data set from a track hub, so UCSC provides track hub configurations. And if you know one of these track hub configurations, you can put in the link to the track hub configuration and we'll automatically import all the data sets from that track hub that are part of that track hub. And then you can create tracks on Epivis within the NEMO platform. But if that is not available, you're also free to either upload your BigWig or BigBed files, or you can point to a file that is publicly accessible. As long as it's a BigWig or a BigBed file and it's publicly accessible, Epivis will be able to visualize the data coming from that data set. And that's how all the profiles that we created on NEMO Analytics are created. Those are files that are sitting on NEMO archive. We did not download them and re-upload them to NEMO Analytics. They're just pointing to data sets that are on NEMO archive. And once you upload or link to a file, you can you have an option to name the file, which I think brings back to the previous question. We use the same convention that was in NEMO archive for the files. You can also add any annotations for that particular file. Every file that you upload needs to be associated with the genome build. And we can definitely add MOS. We currently support the MOS MM10, the human HE19 and the HE38 versions, and the marmoset dataset. So if you need more genomes in this, please let us know and we can definitely add those. But every dataset needs to be registered or linked to a particular genome build so we can show the appropriate genomes or gene tracks when you try to visualize those. One of the important key features of NEMO Analytics is choosing a permission on who can access these files itself. So if you choose the file to be public, everyone on the platform will be able to access the file, but you can also choose this file to be private, in which case only you will be able to access the file and explore these data sets. And once you set all of these, you can upload multiple files or you can submit, and that would register all these files on the platform. So that's the upload part. And once you finish the upload part, there's another link in the drop down called the Epivis Panel Designer. And if you go there, this is what lets you create that Epivis panel that Brian and I was showing you before. You can add a title to what your panel could be, a description, a long description, and what data you're trying to show or where these data sets are coming from. You can also choose an access level to the panel itself. If you choose the access to be panel, anyone on the NEMO platform will be able to add this panel to their profiles. If you choose this to be private, only you will be able to access it and add it to your own profiles. It's also important that you choose what genome build to use for the panel itself. And that goes back to how how the gene search works within NEMO Analytics and how we can search for the location of gene within this genome and show that region. So once you configure all those parameters, you'll see the panel itself. There's an add chart feature where once you upload all of the files, you're clicking on add chart will show you all the files that you uploaded. And once that's in place, you should then see if you save the panel, that gets stored to the NEMO Analytics. And you can make all these customizations about settings and colors, and the way you organize these tracks or visualizations is persistently stored within the NEMO platform. And I think that brings back to the end of my presentation. And Brian will show you at the end of the presentation how you can add these panels to a profile.
All right, thank you very much, Jairam. So you should be seeing hopefully the workshop panel again. I don't see the little blue or the green outline to my screen. So
Yes, it's showing your
Excellent.
Google Doc. Yep.
okay, wonderful. So what we're going to do is I'll take you on a quick walk through the single cell workbench, but we probably want to spend a little bit more time on the last portion of building profiles. So I'm going to be jumping through and showing you kind of like, you know, as the classic cooking class, you mix everything up and then there's already an example in the oven. So I may be just jumping to that final example. But it might be worth taking just a few minutes walking through how to upload data. As you saw from Jairam's presentation, the epigenetic data is handled a little bit different than the transcriptomic data. With most single cell data sets, you have at the heart of it a gene by cell table, and you also have associated metadata along with it. So we give you an example here. If you click on these two files, there's an example metadata sheet. And this has just a couple very simple fields. Anything that you see highlighted here are the required fields. And so you have a title for your data set. You know, make sure that that's something nice, long enough that it's unique and you're able to recognize it once you get back into the system. As I mentioned before, there's already hundreds of data sets on the system. So if you're adding your own, make sure it has a nice unique name. And the summary can be a little bit longer, so this will be added into the info section there. And the data set type is important. So if you download on the site itself, let's see here, if you go into, let's just go to the front page a moment. This section on uploading, in that upper right, uploading a gene expression data set. This is essentially the front page for the portal for adding in single cell data. So if you needed that example XLS, both the one that I gave you and if you need it as well, you can download it here. All right, so let's take another example, look at what that is. There's a certain set vocabulary of different types of data set that you can use. And then this is the only real tricky part of this. In order to keep the genes straight on NEMO Analytics, we actually link them back to the ensemble ID. So we try and make everything unique. The difficulty is a lot of times you may have different isoforms for the same gene, and within your data set, it may be multiple entries for like SOX2 just based on all those different isoforms. In order for our platform to work, it has to be uniquely tied to a single ensemble ID. So one part of generating the data. I'll show you what's unique about it, but you have to have that lookup table of ensemble to whatever gene ID you care about. And you also have to give the ensemble build. So that's really, in my experience of getting data ready, that's kind of the trickiest part. Then you have a little bit of the contact information. And then the last thing that's kind of important is the tax ID. So this particular data set, we're mimicking it being a human data set. So that's 9606. I think for mouse, it's like 10,020 for that tax ID. You can go to NCBI and look up those tax IDs. All right. Now, the second portion of this, so that's the separate Excel sheet just to prep the system to know that the data is coming. The second one is this is an example of how you might upload the data. So one way you can format it is also in Excel format. And you'll see on this Excel spreadsheet, if you can see down here in the bottom left-hand corner, there's that expression table. So this is just simply genes in the rows and cells in the columns. So we have that unique ensemble ID. And then you have each individual cell, either based on a barcode or however you're labeling your individual cells. So that's that entire gene matrix. And you'll notice right here that it's a dense matrix. So we have all the zeros in there. There's other ways for larger data sets of moving it into a MEX format, a tab delimited format. So you do have more than just simply this Excel option. I'm using this for illustrative purposes, and since this is a small example data set. For the gene tab, this is where I was telling you about the lookup table. So you have the ensemble ID to, in this case, that gene symbol. So that is like the only thing that's kind of critical in this. And note the column headers. That's what they have to be for it to be inputted. But again, that's really the only tricky part right there. And then for the observations, this is going to be the cell level metadata. So each column header, each one of those cell IDs, you can associate with it any and all metadata that you want. This is especially important if you're publishing a paper, you want to build a profile. Perhaps you've already have all the clustering information. So in this case, you see that we have our t-SNE coordinates that could be read in and used in that profile. If you have the cell type or what you think the cell type is, you can list that there. Different cell classes, so you can have multiple resolutions. So you can maybe just present your data as neuron, non-neuron, then excitatory, inhibitory, and then get into the various cell types. That's all possible there. If you also have this information, that means that analysis can be picked up in a single cell workbench, and then folks can analyze your data from the point of view of actually having the cell types clustered already. So that's a huge benefit to anybody looking at your data in the future. So that's the kind of breakdown of how the data has to be formatted. And I'll just show you how that gets uploaded. So if we go to the NEMO site, and again, how I got there was in this upper right-hand corner, clicked on gene expression. And the first thing you do is upload that metadata file. That's that Excel file. So these are the two that I downloaded from this presentation. Again, that's just going to be that XLS. And it reads that very quickly, and you can just simply double check all your fields, make sure that's what you want. You scroll down to the bottom and you click continue. And once you're here, you're prompted by the different types of, or different formats for the data. So again, I just walked you through this Excel. You can also do a MEX file, which is more common to what you might see coming off of Cell Ranger or other alignment pipelines. That'll be, again, broken down into 3 files, one describing the data matrix, one describing the genes, and one describing the cell observations. And then the other way is a 3-tab tarball, which is actually something that I've used for very large data sets. It simply, again, stratifies it into counts, genes, and cell information. And then within each one of those things, you can actually have a sparse matrix for this one and add it in there. Okay, so I'll select that expression file. In this case, that cortex data. I can open it up And because this is a small data set, it'll just take a few seconds to load up, hopefully. And again, it'll summarize what it's found. In this case, it is only 94 genes by 160 observations. And we submit. Now, once you do this, you're actually presented with the option of plotting the data set. So if this is something that you already have, those cell types, you may jump right into creating plots. If you didn't want to do that, if you wanted to actually use a single cell workbench, you can simply go back to the home page and go into the single cell workbench. So we're going to do that now. Jump to the front page. And we see that tool here on the left-hand side, the single cell workbench. Okay. And from here, your list should probably look a lot simpler than mine. So these are all the various data sets that we're working on under the curator data set. And you should see right at the top, hopefully, that same data set that you just uploaded, the Cortex example for workshop. But this is also a nice way of, if you wanted to get into any of those other data sets on the system, you can go back and reanalyze it. Now, again, as I mentioned, for time, I'm just going to describe this, maybe show you a couple examples of what the outputs are that you expect. But again, you can do all types of filtering like you typically would in a single cell experiment. Sometimes you want to exclude cells that don't express many genes or express too many genes. I know that kind of sounds odd, but these might be indications of doublets where it's just more genes detected than you would expect. You can also filter based on the genes themselves. So if you find the genes are only expressed in a handful of cells, perhaps that's spurious. If they're expressed in many, then that might also throw off the analysis. So you might want to filter these guys down. Then as you go through the various steps, you can QC based on mitochondrial content. You can identify those variable genes. So you're reducing down your data set, and then you can do the PCA and bringing it into reduced dimensional space. So instead of going through this exact workflow, I'm just simply going to show you those different steps. So we were saying by this is an example of that output for the filtering. So what you would see is some of the top expressed genes. This may help you identify if there's a gene that's really high in absolutely every cell. That's where you could exclude that by simply saying, okay, get rid of those genes that are in more than 90% of my cells. Because things like MALAT1 a lot of times will skew the analysis a bit. So I mentioned before that you can then filter based on mitochondrial content. And what that looks like is, again, you can grab that gene, if all of your mitochondrial genes start with like MT or something like that, you can select that. And then you can see from this what kind of parameters you want to select. Usually we filter by about 5% mitochondrial content. If that's very high, that could indicate either stressed cells. So as they're moving through the sequencing process, the mitochondrial content just got thrown off. Then selecting variable genes in your example. So again, a lot of times you don't want to do the analysis on all of the genes. Most times you'd be doing a selection of like 30,000 genes, and you may pare that down to like 2,000 genes. So under this selection, you can actually select out a couple of different ways of identifying those variable genes, or you can simply say, give me the top 2,000 variable genes. So there's a couple of different ways you can go about and identifying those. So once those variable genes are selected, you can go in and do the principal component analysis step. And so if you desire, you can select the top variable genes. And what it'll do for you is it'll show you what portion of the variance is that PC explaining. so you can select down how many PCs you actually want to use. And then lastly, clustering the cells into a couple discrete groups. So in the case of the our data set here, again, it was just a couple of cells themselves, astrocyte oligo. And this will be the first identification of whether or not you have good clustering. I'm going to take a quick example. Let's see if we can dive into it from here. I'm going to try and see if there's a data set that we already have analysis for. All right. So if you've done all those various steps, so for a new data set, you would go through that, select the mitochondrial, find the variable genes, do the PC and UMAP. Again, the clustering, you can actually have control over what resolution of clustering. And so once you feel like that's a proper resolution, then you can move on to finding marker genes. So what I'm simply doing here is that I'm taking one of my data sets that I've actually already done analysis on, and I brought that in, and I'm simply taking advantage of that stored analysis. So again, under this section here, you can actually go through and do that new analysis, or you can use something that's already been inputted. And so how this looks for finding marker genes, say I wanted to find the top 10 marker genes for every single one of my clusters. Oops, I'm sorry about that. All right, I may simply show you this from before. All right. So an example output from there, again, in this case, we had 5 different clusters. You would get those top genes from each individual cluster. You have this described in chart form, and again, you can simply download this as an individual file, and you can follow up and use any of the web tools if you wanted to try and identify what the cell types are if you didn't know them already. Okay, and then this last one is a nice comparison tool for any data set that's on the system. And so again, I think I was showing you this before. At the end of that whole process, you can select 2 different clusters and then identify differentially expressed genes between any 2 clusters. All right. So any quick questions on, let's see here. Quick questions on the single cell workbench before we jump into profile building? Okay. So this last section, you can explore a little bit. If you did process the Cortex example, you can try that out. We're going to go to the dataset manager and try and create a new profile. So again, we'll just go to the front page here. And this is going to, we're again going to the top right, and we're going to click on Dataset Explorer. So any data sets that are on the system, you can simply type in any search terms that you desire. And again, you can say something as broad as human, or you can type in a certain brain region, see if that picks up. You can select by data type. And so within this, you can do a couple of different things. So you can take a look at the information associated with a particular data set. And again, all of this is editable. So if you're uploading your own data set, you can edit that information. For the display, you can go in and curate. So this is something we're going to do next. You can share the data set. Each one of these data sets has their own unique link. And then you can edit that. So what I'm going to do with you right now is walk through a couple of different types of plots that you can make. So for a given data set, you'll be presented with this. And again, this is under the curation view. And here we have a choice between primary data and stored analysis. So if this is a data set that you already have brought in, identified different cell types, that is going to be under the stored analysis. Let's see if we have that for there. Primary. Okay. Oh, I see. I'm terribly sorry. So stored analysis will be a new from the single cell workbench. For primary data would be what's already uploaded. So again, we have a couple of different options here. So we have the scatter. The t-SNE static is those views that I've been showing you up to the now, where you have two different plots right next to another. One is showing the the genes, and then one is showing the cell types. Dynamic is just purely the gene expression. And then we see bar type, violin, and line, like we've seen in a couple of different profiles. So I'm just going to show an example of a t-SNE static. Once this becomes available, you can type in any gene that you desire. This is not something that's permanently fixed to this plotting, but it's just a way of trying out and generating. And then the display name is optional. Let me try and find one that has a nice primary analysis. So say, for example, we're in the front page right here, and we wanted to make perhaps a violin plot of this. So again, we can go into the curate for a single data set. And as we saw right here, there's already a few different plots that have already been generated. If we wanted to simply add a display, again, this will show a little bit better what I was trying to get to in that last one. Just make sure that that exists. There we go. So how this might work with primary data that you've inputted already. When I'm selecting down here, these are those things in the cell type metadata. So when I added in the t-SNE plot coordinates, that's where those guys live. So I can simply do t-SNE1, t-SNE2. If you're trying to recapitulate something that's already from a previous analysis. And so there is that exact plotting that you may see in the paper. If I wanted to show a legend right next to it, I can click on Show Colorized Legend. And then this is where I can select whatever level of clustering I want. And it's just a slightly larger data set, so it'll just take a little bit more time. Oh, that might be, let's try class label instead. Would be better. Okay, that's much better. So we see that this is actually a slightly different resolution of the data than what's presented in the profile. So again, this was just simply another metadata column. What we have in the profile is a little bit more granular. This is just simply at the level of higher level neuron classes. And so this can be simply added as another one of these plots, and that can be created as a default. For a violin type plot, so again, we can take a look at, say we do that for the violin, at 2. And again, this is now where we have a lot of choice with how we present this. So along the x-axis, this is going to be how we stratify those groupings. So if I wanted to do the exact same thing, class label, let's say we do that, and then y would simply be the expression of that gene. If you wanted to have a specific color for each grouping, you can, but let's take a preview of that. And there we have those same groups that we just saw in the t-SNE, but now broken down by violin. And so you see, it makes good sense. GAD2 very expressed in the GABAergic populations. And then this could be a plot alongside other things in your profile. So hopefully for this small portion right here, we've gone through a couple of different ways of plotting the data. Jay Rama showed you how to do the same thing for the EpiViz panels. So now that you have an idea of both uploading data, presenting the plots, how do we put all this together in a profile? All right, so we go back to Dataset Explorer. And this is kind of a slightly hidden thing, but one thing that's collapsed to begin with is something called profile management. And this is where we're actually going to be building those profiles. And so for the ones that are established, for example, the mini-atlas transcript, we can take a quick view of how this is presented. Currently, I have 5 different data sets in this profile. And for each one, we can do all those same exact things. If we wanted to change the display, we can. If we wanted to remove them from this profile, we certainly could go through and do that. So one nice thing about also each one of these profiles, if you wanted to share this link, want to share this profile to either one of your collaborators or have it listed in the paper, this little share layout profile, just clicking on that copies the URL. And then that, if you put that into the web browser, it simply takes that profile unique identifier, and that's that short URL that you can use to your paper. And then that goes right there. So now one extra little added portion of that is if you take that URL, if you add in a gene of interest. So if you say and G equals, say like that GAD2, then what it'll do is it'll jump right to the profile and display that gene directly. So hopefully that gives you a good bit of flexibility for sharing these profiles as well. Okay. So then how do you make a new profile? So if you went to simply adding to the, clicking on the plus sign here, and if we made up a new profile for your study, say like, I'm just going to put in test workshop, going to add that. And now what I can do, this kind of things, these for whatever reason stay, but I would start by trying to identify what data sets I want to put into this profile. So again, maybe I'll type in human cortex. And again, the whole host, there's again, hundreds of profiles or hundreds of data sets that are on the analytics already. Say I simply want to, maybe I'll add in this one, maybe I'll add in maybe this one, maybe this one. So again, these may be data sets that you uploaded that were already on the system. And so let's see what I've selected. I have to do a hard reset on that. Okay. There we go. If you find that every single data set is in your profile, just simply refresh the page and find that profile again. But these are those three profiles that or those three data sets that I've chosen. You can, once you have that and you're trying to understand how you're going to display that data, if you click up here at the top to the arrangement view, this is where you're actually going to be organizing how that page looks. So what I'm going to do is I'm going to actually, you can expand these to be the full width of the website, or you can have it just side by side like this. You can move them around. So if I wanted to have them displayed like this, you can. So we're going to do that, and it's important to click Save. And now once that's arranged and you have all the plots that you like, what you can do is go back to the front page and try this thing out. So I'm going to find that one that I just built, Test Workshop 2. I'm going to search on that. And now this is my new profile that I've just built. And again, this may be meaningless because I just threw a bunch of things together. But essentially, if you don't have a plot for this built yet, you can go in, curate that data set, create a new plot, rearrange things however you like. But the cool thing is, again, if you have a publication, this is a wonderful way of putting that data together. If you want to use this as a tool of understanding how your data compares to other data, maybe you have your own motor cortex data set, you've put it through the single cell workbench, you've curated it, and now you'll put it next to a bunch of other data sets and see, oh, well, okay, this gene is, sharing this particular cell type. Or you can use that for identifying your own cell types compared to the other data sets. Here's just an example of a profile. Let's see here. Of a profile that we built very recently on human developmental data. And again, just a bunch of different views of that data. So the nice thing is that, again, you can have this mixture of things that really dissects, in this case, different neurons of the different regions of the hypothalamus in this case. And really at a snapshot, you can see exactly how that gene expression is stratified across the various regions. And hopefully the types of plots that are available here will give you enough flexibility for displaying the data and how you like. Okay. All right. So we can pause just a moment if we want to have any live questions as we wrap up this portion of the workshop. Or perhaps all the questions were handled in the chat.
Thank you, Brian. Any questions for Brian or Jairam? All right. Is that, and you finished? Is there anything else you wanted to say, Brian, or?
No, I think that's it.
Okay.
I mean, I think just the long and short of it is that if you have any issues with New Analytics, please feel free to reach out to us. There's a lot of talented folks that are both able to help you with data curation and profile building. So don't hesitate.
Excellent, thank you. And I'll just note, Sean has put a link in the chat for Nemo Analytics contact page for future questions or comments. All right, this brings us to the break portion. So we're going to go on break. We will resume again at 2 o'clock with the remaining presentation. So we're on break now until 2. See everyone in about 20 minutes. And we will resume with the remainder of our presentations on various tools associated with BICCN. And next up is a presentation on the BrainOme resource. And our presenter is Fangming Zi. I apologize if I pronounced that wrong, Fangming.
It's good. It's good. Thanks.
Okay, you're welcome. So I'll let Fangming take it away. Thank you.
Okay, can everyone see my screen well?
Yes, it looks good.
Okay. Hi everyone. Welcome to the workshop, and I'm gonna use the next 30 minutes to introduce you, to give a brief demo and introduction on the BrainOme genome browser we have constructed. And in addition to myself, I think we along Galber, let me first make this bigger. So along Galber is my lab mate, who might or might not be here. I guess he's Along, are you here? I guess it's not on here yet. And we are from Iran Mukamal's lab at UCSD. So it's actually nice that we have a small group of people today, and that means we can make it as interactive and interruptible as possible, as you guys would like. So please interrupt me anywhere, whenever you want, and if you have any questions, I would be very happy to answer it. And I plan to jump right in to give you a view of how this tool look like, how you can use it, and I encourage you to follow my step as much as you can as I'm doing it. And if you don't it's very understandable that you don't completely follow throughout the way. And that way, I also prepared, in that case, I also prepared a brief README file like this that you can follow on at your own pace. So let me first share this document with you. Anyone, okay, can comment, that's good. I'll share it in the chat. So you can access it, and it contains everything I will be covering today. So that's the overall logistic of my next 30 minutes. Is there any general questions? Let me just pause for a few seconds in case there is one. I see some anonymous came in. Okay, 3 people, 4 people, that's great. That means you already have the access to this file. Okay, so let me just jump straight into the browser we have, and the link is in this document. It's brainome.ucsd.edu, BICCN_MOP. Let me just start from scratch and type it right in my browser. This is my Chrome, brainome.ucsd.edu, then BICCN_MOP. And then you come to this browser, and this is the entry page and the default view of this browser. And I'm going to show you step by step. First of all, what it is. It's obviously a genome browser, but what does it contain? And also what functionality it offers and how you can use it. And in the end, I would like to showcase how we use it to do our research. Okay, so first off, what it is. So there are 2 good places you can find resources to learning more about this browser. One is from the entry page of this browser. And if on the top panel, you have a couple buttons, and there is one about right here. If you follow, do you see my annotation, some green circles? Yeah, if you click on that button, let me first remove everything. Click on this about, that takes you to a page that gives you an overview of the interface of this browser. It's called NOJ. It's not developed by me. It's developed a few years ago by a postdoc, by several person from Joe Ecker's lab. And this is one place. Another place to learn about this browser is from our paper, and the link to this paper, to the bioRxiv version of this paper, is also in this document. And the paper is accepted by Nature, and it will come online soon, I think next week or so. But let me just take you to that paper and go to the first figure in specific. And that gives you an overview of what this browser is about. So essentially, we surveyed a specific area in the mouse brain, primary motor cortex. And this is where it is in the mouse brain. And we analyzed 9 different data sets from mouse MOP that contains RNA-seq from both the single cells and single nuclei, and also epigenetic sequencing, including open chromatin attack-seq, and DNA methylation, or SNMC-seq. And this is an overview of how many cells we have and how many reads per cell that contained in those data sets. And the browser I'm going to show you display the result of our analysis in terms of the cell type specific profiles of this of molecular informations like this, like panel F of this paper. So here is one example at this gene TAC1, and for a specific cell type here, it's called parvalbumin reelin _ KELB1. It's one cell type from that part of the mouse brain, and it contains 3 types of information, actually 4 different types of information in this example, which including DNA methylation, we call it MCG. It's the methylation at CG dinucleotide context, an open chromatin or ATAC-seq like this, and also 2 types of RNA-seq profiles, including the ones from single cells and single nuclei. Some of you may already be familiar with genome browser, and some of you guys may not. Let me just briefly explain what they are, and if you have more questions, I will be happy to answer. So on the top track, it's what we call a gene annotation track, or gene model track, which shows you on the linear genome where the locations of every genes are, and we happen to show you the location around one gene, this TAC1, and this arrow shows you the transcription direction of that gene, and the 5' end, 3' end, etc., etc. And exons are highlighted in green, whereas other parts of the gene, introns and other parts of the gene body, are not highlighted. So if a region is not covered by a gene, it's intergenetic area, and it's not belong to any gene, but it's still part of the DNA. So then follows the molecular profiles, and the first track shows you the DNA methylation profile, and it's green, it's colored by green, and it contains a lot of I would say bar plot, and where the bar is very narrow, and each tick shows you one genomic location, one CG site along the genome, and the height of the tick represents the DNA methylation level, or DNA methylation level means the fraction of CG site at the fraction of CG we captured from that site that are methylated. It's a value between 0 to 1. 1 means that site is completely methylated, and 0 means it's not methylated at all. And as you can see, there are ups and downs on this tracks. So here it means it's a hypomethylated region or low methylated region, and here it means it's a highly methylated regions. And likewise, the next track shows you open chromatin profile or chromatin accessibility profile, where the peaks shows you the open chromatin, and if it's not a peak, it's either open chromatin not captured by this assay, or it's a closed chromatin. And the next two tracks show you gene expression, and as you can see, gene expression can only happen within gene body. I mean, therefore, there's no signal outside of a given gene. So that's a good overview, and it's nice, and it's curated in this manuscript. But what the browser is able to show is that it shows you many more genes, and you can customize it as you like. So another way of entering this our browser is by click on this paper, this URL that carries you directly to our browser as well. So then you can use it and manipulate it. So let me see where we are. So I just briefly introduce you what this browser is about. I'm next going to show you how we can use it. Okay, so let's start from scratch again. So just in case you didn't follow the first time, I'm going to do it all over. So this is the default page of this browser, and as you already familiar, and there are a few tracks. In the top track is gene models, and here's our few tracks of DNA methylation, and followed by a few tracks of ataxia. Now the question is, how do we navigate through this browser and use all of the features, right? So first of all, you can jump to any of the genomic location as you want. There are several ways doing it. You can just scroll and drag your mouse from left to right, right to left, then you can scroll through the whole genome manually like this. And if you have a particular location that you want to look at, you could type in the genomic coordinates. So, and that is on the top panel of this browser, where you have a location menu. 1 means we're on chromosome 1. And you can choose from 1 through 19 in XYM, because this is mouse genome, it has 19 autosomes. So let's say if we want to go to location 12, and the next variable is the coordinates, where they are on that chromosome. I'll just keep it as it is and then click go. Then the whole view will go to that particular genomic location. Now we see nothing. That's probably because chromosome 12 don't have that many base pairs. So that's my hypothesis. Let's remove one, go. Okay, now we're fine. Now we see some profile right at this location, chromosome 12, this much coordinate. That's one way of navigating through the genomic location. Another way is by searching a gene. For example, GAD2 has already been shown in the previous example. And you can type in gene and search for genes right here. I did it too quickly, probably. You could type GAD, then all the genes with name GAD in it will show up. And then you could type in GAD2, then it leads you to the location where GAD2 gene is. And yeah, another thing you could do is to zoom in and out in the genomic location. And to do that, you could manipulate this zoom level panel. You could zoom in by click this button, or zoom out like this. Okay, and you can also specify some value here. If you don't want to go to 400, you can type 300. That does the same. Okay, I think that's pretty much how you can navigate through different genomic locations. And that, obviously, another thing to do, you want to do, is to navigate to control which tracks you want to show, right? So each track is a specific data modality for a specific cell type. Like here, so for this particular track, it says MCG. First of all, MCG means it's DNA methylation and CG locus. And then the following strings specifies which cell type they are. LAMP5 means it's a cell type with LAMP5 positive gene expression, and it happens to be inhibitory neuron. And LAMP5, PDL-LAMP5 is a subtype of LAMP5, and it also have some string C6_4 is just a label we gave to that cell type. And you can see that in the ataxic panel, that same cell type is also shown here. That's because we aligned them together. But you can control all of these features by selecting on the panels right here as I'm pointing to. So here you could choose cell to show. You could want to show all cell types in MOP, or only excitatory cell type neurons, or inhibitory neurons, or MCG derived, CGG derived, or none of them, or you want to select, you want to generate some custom tracks. Let me just give you a few examples. For example, if you want to select inhibitory neurons, you can do that. Cluster depth means how granular you want to show different cell types. We know that cell types are organized in hierarchy, and you can have broad level cell types and fine level resolution. Level 1 is broad, level 2 is finer. And modalities to show, we here clicked MCG and ataxic and enhancers. I'll talk about that in a second. But you could also show RNA-seq as well. So if I click scRNA, single cell RNA, and snRNA, single nucleus RNA, they will show later. And you could also choose how you want to arrange your tracks. You could either arrange it according to cell type or according to modality. Let me just show you one way and give you a few. I'll choose arrange by modality. And once you set it up, set them all up, you can select reload browser, and they will automatically reload and generate this new view for you. And now you can see that the panel, now the panel showed all of the inhibitory neurons tracked with 4 different data modalities because we selected them. That's again DNA methylation, ataxic, RNA, single cell RNA, single nucleus RNA. Yeah, but but this is just one example. You can choose you can arrange it in other ways. For example, if I arrange it by cell type and click reload again, now it's displaying the same amount of tracks, but we reordered them so that tracks from different cell modality but belonging to the same cell types are organized together instead of previously different modalities are separated. So that's how you can arrange them, but these top tracks provide you with how we typically arrange certain tracks and visualize them, but you can also customize it as you like. For example, if you want to move this second green track, MCG LAMP5 SLC35D3 up to the top, only that track up to the top, while keeping all of the other tracks in the same order. How do you do that? Well, to do that, you can go to the left side panel, then find the tracks you want to toggle with. That's here. So going from the up to the bottom, this one corresponds to the track over here. Then you just toggle it up to the front. then it jumps to the front. So that's how you can arrange your views in different ways. Okay. Of course, another useful feature I really like about this is so once you generated the configuration, for example, if we want to focus on this GAP2, maybe I'll choose a different gene, for example, cuts SATB2, for example. So now it's loading. Once you've generated this view, okay, and you've customized everything. If you want to share this view to your colleagues and to present it somewhere else, how you can do it. One way to do it is, of course, to take a snapshot, screenshot of this view, but it's not that helpful because people might want to look in the neighboring area. They want to manipulate it themselves. Another way is you can click this copy shareable link. And if you do that, and you can share this link to everyone else who might want to look at this view. And that link contains which the tracks you've configured and the genomic locations you selected. For example, since now I've already copied this link, I could go to, let me pretend that I'm now a different person with this new link that someone has shared with me, and go to that link. That directly guide you to the location we were looking at and the cell types we configured. So that's I think pretty much how you can use this. Oh, another important feature I forgot to show you is so sometimes let me first show you all of the tracks, and by selecting only a few cell types so that you don't feel overwhelmed. So sometimes you feel like the peaks, the height of the peak is not large enough. It doesn't take advantage of the full view of your browser space. And so you want to adjust the height of your tracks. How could you do that? You could click here, try track height up, and they all blow up like this. That way you have a clear view. Sometimes you want to collapse them down. You can just click like this. Okay, and also sometimes you don't want to adjust the track height. Instead, you want to renormalize the tracks such that the peaks are all more obvious. So to do that, you can click the button here. Then all of the peaks will grow up. It's just renormalize every track to renormalize the height to show it larger. And sometimes when it's too high, you couldn't see the contrast and a lot of noise show up. That's because many of the lowly expressed, not lowly, lowly accessible in the case of ataxic regions are blown up because you normalize it too much. And then you can bring it down like this to better show the contrast and where the highest regions are. So for different up and down buttons you can play with, as this goes up, this goes down, this goes up, this goes down. Yeah, I think that covers pretty much everything. And let me see how much time do I have? I have 6 minutes left. Is there any questions up to this point? Let me just pause for a few seconds. Okay, so if not, I'm gonna next show you how we are using it in our research and in our paper, just to give you an idea. Okay, so I'm here going to, again, to the paper I shared with you. And instead of looking at figure 1, I'm now going to figure 4, where we actually showed a screenshot of the browser we just shown you. So I'll probably show you how, as an exercise, how we can come to this page and what interpretation we have about what do we learn from looking at this browser chart. So on the left panel, we're near we're in a gene location near the transcription start site of a particular gene, TSHZ2. And we are showing you 3 different data modalities and a bunch of different excitatory cell types, and including the IT cells from layer 2, 3 up to layer 6, and CT cell layer 6B, PT, and MP. And you see that around that area, you have a specific low DNA methylation and in the MP neurons, low DNA methylation highlighted here. And correspondingly, you see only high ataxic signal at MP cells around the same region, but not in other cell types. And also you see high gene expression only at those MP neurons, but not for other cell types. This means at that certain gene, you see a nice correspondence across 3 different data modalities. The low DNA methylation corresponds to high ataxic P corresponds to high gene expression only for MP excitatory neurons. Okay, so how could you go to that site? One is we already provided with a link to you. You can just click that link. Another thing to do is you can go to the browser on your own and select, for example, excitatory neurons, level 1, and show DNA methylation attack and single-cell RNA-seq and group tracks by modality. Okay, and then let's go to that particular gene. It's loading. It might take a few seconds. TSHZ2. Oh, it's already here, but if it's not, you can just type that gene. It leads you right to this page. Now, now the browser seems to be a bit different from what we've shown in the manuscript, because in the manuscript we customize the order of the tracks, but you can also do that as well. And you can already see that you see high DNA methylation throughout the region, except for these 2 tracks. And the first track happens to be an NP cell. It says L5NP. And the second track is also from NP cell type. So it's consistent with what we've shown, but the orders are a bit different. Those 2 similar cell types are in 2 different locations. So just to apply what I just shown you, how do we bring those 2 cell types together? It makes sense to bring them together, right? So to do that, you could try to find on the left panel the second NP cell types, track for the second NP cell type, which is right here, and toggle it up to have it next to the other NP cell type, which is right here. If you do that, you bring the 2 tracks together and they all look the same. Also, although it does involve some manual work, it always takes some time to make it look making sense and straightforward. For example, that does brings the MCG tracks together, but it doesn't bring the 2 separate ataxic tracks together. To do that, you can do the same thing. You find NP tracks in the attack panel and bring it up. Now you see the 2 peaks are close together. Okay, so that's an example of how we are doing it, and I think we're about on time. Let me just ask again, is there any last questions you guys have? And I'm always happy to help to answer questions even afterwards, but in case you have questions here, you can ask about it.
Thank you, Fang Ming.
Yeah, thanks.
Questions, anyone? Yep, here's one in the chat.
Okay.
Oh wait, no, we already got that one. So nope, I don't see any new ones in the chat. Anyone, you feel free to just unmute and ask if you have a question. That's totally fine.
Just to give you a few more example, and this is the manuscript, the bioRxiv manuscript of the BICCN MOP flagship paper that contains 300 authors, more than 300 authors, as many people are here today. And if you go to which figure, let me see, figure 8 of this manuscript, and panel A shows you a t-SNE or UMAP embedding of all the cells. For panel B, you see there are screenshots of this browser charts, and they show you several different locations throughout the genome: COX2, RSPO1, HTR2C, RRB, and 4 different tracks, types of tracks, and a bunch of different cell types. And just another example of how you can visualize it and how you can use it for your research. And you see nice corresponding and layer-specific structure of the molecular signatures across these cell types. Okay, so I'll just stop from there. Okay, thanks everyone. Let me know if you have any other questions, and I'll be happy to answer them either in chat or send me an email. You can use this tutorial for your reference.
Sounds great. Thanks very much, Fang Ming. We have a couple more minutes if anyone has a question. And we will send these links out in an email to everyone too, just in case we lose them after the Zoom ends. All right. Okay, then we will move on to the next segment, which is a presentation on Catalyst, and that will be by Yang Li.
Thank you, Michelle, for the introduction. I'm very happy to be here to share the resource we have, and it's a really good opportunity to have all you here to using our browser and also helping us to improve. So first of all, I'd like to share a Dropbox link, which is a PDF file, including the data I want to show you today. So if you find any questions, you can always go back to that file to reference. Let me first share my slides. Can you see my screen now? No?
Oh, it's coming now. Yep, we see PowerPoint. Yep, perfect.
Great, thank you. So first of all, I'd like to quickly go through the data set we have and what is the usage of this data set. So what's the question you can ask using this data set? And for the second part, I will go to the browser and guide you to explore the functions. So first of all, I'd like to introduce our data set. I know there's a lot of fantastic resource to explore the RNA expressions, but there are a few data set allow you to explore the epigenetics landscape for all the neurons. So and also we are quite interested in to answer how the gene expression is regulated by the cis regulatory elements, for example, the promoter and the enhancer. And we are also quite interesting to link those enhancers to the putative target gene so that people can using that resource to do some validation or to do some research on their favorite genes. So to do so, we need some of the high throughput data set to map those cis-rare elements. For example, the taxic data down by the encode. So our approach here is clearly is the single cell taxic data developed by RenLab and by Sebastian. And later with the help from Xiaomeng, we generated a lot of single cell taxic data from the mouse brain. And as a lot of people already mentioned, we generated this data in adult mouse brain. We basically collected more than 800,000 nucleus from 45 brain regions. And they are basically, all the regions are in the mouse brain cerebrum, including esopcortex, olfactory area, hippocampus, and cerebral nucleus. So from this data set, we basically annotate 43 major types. This is annotated based on all the regions. In our paper, we are not quite focused on the MOP region, but you can still looking for the cells from that region as well. So from our data set, we basically saw 1/3 of the cells are non-neurons, and another 1/3 of them are GABAergic neurons. And the last 1/3 of them are glutamatergic neurons. And all these 43 major types are annotated based on at least 3 marker genes published before. I will show you a genome track here, so you can see like those are several marker genes we use to annotate those cell types. And for each major cell type, we also did a subcluster in order to find more detailed subtypes. I will show you one example here. If you look at the SST GA, which is represent the SST positive GABAergic neuron. If we do this subcluster for this major type, we will find 10 subclusters. And there's one of them have this chordal gene accessible. Which possibly is the SST chordal cells. So those cell type we annotated are also confirmed with other modality, for example, the methylation data and the single cell RNA-seq data. It seems like we capture all the major types from the single cell RNA-seq data, and they aligned pretty well with previous published taxonomy. So another way to use of this data is to explore the regional specificity. So as you can see from the middle panel here, lots of the cell types are quite specific to one of the region. Of course, for example, the granular cells are very specific in the CA and DG hippocampus, but also you can see there's a gradient difference across the whole cortical region. And we find the exciting neurons have a higher regional specificity, as well as some of the GABAergic neuron. But surprisingly, we also find there's a regional specificity or gradient difference for the non-neural cells. So here is the overall overview of all the subtypes we have. And from this bar plot, you can see like which cell type are quite specific to any brain regions. Just a quick example, here is one of the subclusters for PV positive GABAergic neuron. And you can find there's a tiny population quite specific to the striatum, which could be the striatal PV interneurons. And if you use our browser, you can always look into interested genes. For example, here, the KIT, which quite specific to this cell type. I will talk about this later.
Real quick, Yang, sorry. Real quick, there's a question in the chat asking, do you map these cell types to ontologies?
Yes, we did not map those to ontology, but we used the ontology data to confirm the annotation. I hope that answers your question. Okay, so moving forward, so what's the major contribution here is not only the cell type annotation, and I guess there are more cell types can be annotated using RNA-seq data, but the real information we provided here is the candidate cis-rare element, which is the CCREs for short. And among those 490,000 candidate cis-rare elements, we find most of them are quite cell type specific, which means those cell type specific cis-rare elements could be the drive the cell to differentiate or to subtend the cell identity. So those candidate cis-rare elements you can use to explore very interesting questions. I will show you some of the examples here. First of all, if you look at the differential cis-rare element across all the subtypes, for example, here is the SST trodo cell. You will see we identify 7,000 candidate cis-rare elements. They are quite specific, opened in this SST trodo cell. And using those elements, we can find this potential motifs enriched in the cell type. And with the help from RNA-seq data, you can identify one of the TF, for example, here is KLF5, highly expressed, and also have its motif enriched in the cell type specific elements. So we are suggesting the KL5 could be one of the important driver for the cell type. So this is one of the way you are using those candidate cis-rare elements. The other way is we are really interested to link those candidate cis-rare elements to its potential target gene. So to do so, we using basically two approaches. We consider the co-accessibility using Cicero, but also we consider whether the opening of this candidate cis-rare element are correlated with the expression of surrounding genes. So using these two approaches, we are able to identify 129,000 positive correlated pairs between a gene and its potential enhancer against a random background. So here is the heat map I want to show. From the left is the chromatin accessibility values for each candidate cis-rare element. And you can see they are grouped into several clusters, which are specific to major cell types. And from the right side is the expression level of its potential target gene. And you can still see like some of the cell type specific candidate cis-rare elements are kind of associated with the expression of some cell type specific genes. So using this link between the cis-rare element and its potential putative genes, you can do several research. First of all, by using that network between the candidate cis-rare element and genes, you can explore you can first of all construct a network and then ask what is the major driver TF involved in the cell identity or cell type differentiation. One example I want to show here is the mutagenesis of the oligodendrocyte. You can see the driver TFs involved in the oligoprecursor cell, committed oligodendrocyte newly formed oligodendrocyte and melding forming oligodendrocyte and mature oligodendrocyte. Another example is you can also using our data to explore the neurogenesis. Since our data set including the olfactory area and also the hippocampus. So within the subgranular zone, you will find the adult neurogenesis lineage. And from the subventricle zone, you will find the you will also find this adult neurogenesis lineage. And using this candidate cellular element and the gene links, you can also explore the main driver TFs involved in this cell lineage. So the last part I want to show is another usage of this data set is we already predefined the putative links between its target gene and candidate cellular element. I want to show one example here is the neuronal intermediate precursor cell. You can see this small population actually came from two brain regions. One is from the hippocampus and the other is from the cerebral nucleus. We know there's two populations of progenitor cells within this neurogenesis zone. And if you look at the open chromatin for the NIPC from subventricle and the NIPC from subgranular zone, you will clearly see the difference. For example, here we have three regions, could be the potential enhancer to regulate the neural G2, one of the gene quite important to the progenitor cells within the subgranular zone. And you will also find another enhancer could be regulate another important gene in the progenitor cell from subventricle zone. And one of them actually is validated before. This data I downloaded from with the enhancer database. So you will see this enhancer is actually active in the forebrain. Okay, so the last part I want to show is we have this candidate cellular element, and we have this link between to its putative genes. What else we can do? We can also associate this candidate cellular element with the risk variance we find from the human. So by lift over those elements to the human genome, we find a large amount of them actually are quite conserved. And this is an enrichment analysis just to show like some of the candidate cellular element we find actually enriched the risk variance associated with different neurological disease. Okay, so basically we identify a lot of cell types, and also we identify the cellular elements. And building on that, we are predict the potential target for each enhancer. And we also show that this data set is worth to explore to interpret the human risk variance. So to let people better assess our data, we built this database called CatClass. So now I will switch to a website, and let me first of all to copy the link. Okay, I hope everyone got it. So we built this data, we did build this web portal, mainly have three functions. So you can use this portal to explore the cell clusters. And we also offer a detailed annotation for all the cell types. And the last thing, you can use a genome browser to explore the track and also the potential linkage between a cellular element and its putative target genes. And I wanted to thank all the Institute and also the USC single cell browser and IGV. So this browser is building on those tools. So first of all, let me go to the cell browser. So if you click the tabs here, the cell browser, they will link you to another page. So in this page, you can explore the cell clusters in different level. First of all, if you're interested to explore all the non-neuron types, you can click the first data set here. And if you're interested to some subtype of the non-neurons, you can open it and go to different data set. For example, you can go to the subtypes of astrocyte to explore more. But here I want to show you one example is the SST GABAergic neuron. So if you are following me, if you go to the GABAergic subtypes, if you scroll down to find the SST positive GABAergic neuron and click open, it will direct you to cell browser view. And here, those cells are colored based on the cell type annotation. And you can always show this data side by side. For example, if you click the wheel and you split the wheel, then you have two browsers. And in one view, you can reference the cell type annotation, but for the other wheel, you can potentially explore the active the gene in each cell type. For example, here I would like to tap, for example, CHODO, because we know this gene is specific to cluster 10. And if you click, select this gene, you will find this cluster, this tiny subcluster are quite enriched of this gene. And you can always change the colors if you don't like it. For example, here, if I change another color panel, you will see this difference more clearly. And another thing is you can select any cells you are interested in and to compare the accessibility of this gene against two other cell type. For example, if I select this cells, you will see this cell have a more accessible at the gene CHODO against to the other cells. Okay, so this is one of the example I'd like to show. And if you're interested in other data set, you can always go to here from file and open data set. Another data I want to show you is basically the radial glial-like cells. If you go to the non-neuron subclusters and find there's a cluster called RGL, that is the radial glial-like cell, and then click open. I'm sorry. Okay, so for example here, I'm sorry about that. I forgot to close that wheel, but you can still see the subclusters for the radial glial-like cells. So the feature I want to show is you can explore the source of each cell. For example, you can click the left panel here if you want to see where is the major, you can color the cell by major region. For example, if I click the major region, you will see the radial glial-like cell from DG are basically came from, of course, basically came from hippocampus. And the radial glia from subventricle zone are came from, mainly came from several nuclear, but some of the cells could be from the olfactory area. And the little of them, we annotate them here as the hippocampus. That is because there could be some dissection issue, because when we dissect the brain, there could be could be some of the cells are pretty close to this region. So we include them here. And if you want to zoom into more detail, you can always do the sub-regions. And again, you can see there are basically two populations of the neuronal progenitor cells. Okay, so this is all I want to see. I want to say about the cell browsers. Let me go to the cell type. So if you click the cell type tab, they will direct you to a new page. In this page, we basically included all the detailed annotation for each cell type. You can always search for interested cell type by typing some keywords here. For example, if you're interested in the radionuclide cells. And for each cell, if you're only interested in the annotation, I guess you don't need to go deeper. But if you really want to see the more details about this cell type, you can always explore more information. For example, I'll show here is the annotation and where this cell came from, all the QCs and the signal track for the cell type. Okay, so next up, I want to show you the tracks. I think this is a really powerful tool for all the people to explore the potential links between a cis-rare element and its target gene. So here is only few cell types showing here as an example, close to the gene SOX2. But you can always select the cell type you are interested in. And to do that, you just go to the setting tab and unclick the cell types you are not interested. For example, I'm only interested in the astrocyte and maybe the progenitor cells. I want to compare with the granular cells from hippocampus, but I'm not quite interested to the IT neurons or the GABAergic neurons. So I just unclick it. So after you select all the cells types you are interested, if you can go back to the track, and then they will load the cell types you are interested in this cell browser. Okay, so I want to show you one way to navigate to your interested gene. For example, here I want to use the NeuraG2 as an example. You just tap the gene and click enter. They will direct you to the signal tracks surrounding this NeuraG2. So as I mentioned in my slides, there are 3 candidate enhancers are associated with the expression of NeuraG2 in the progenitor cells. So this is the way you can explore the candidate cis-rare element relationships with these putative gene. And I want to mention here is the height of each track is can be directly compared across all the cells, all the tracks. But if you are interested in to change it, you can always find a setting, and you can change the track name, track head, track color, and the data range. Okay, so one last thing I want to show is the download page. I mentioned we did a lot of analysis, including the cell type annotation, the candidate cis-rare element identification, and this links between potential enhancer with this putative gene. You can always find this resource here because we provided the download for all the resource. You will find those information from supplementary tables, which I listed here. And you can also find some additional information, including the differential peaks between cell type or between regions. Yeah, I hope this is helpful, and we are very interested to continue develop this web portal to include more data, and we are welcome every comments or suggestions. If you're interested in the data set or have ideas helping us to improve, just don't hesitate to contact me, and you can find my email address here.
Thanks very much, Yang.
Thank you.
Does
Yeah,
anyone? Oh, sorry.
Yeah, this is pretty much it. Any questions?
And again, people can feel free to just unmute and ask their questions if they'd like to.
Okay, if there's not too much questions, I will stop here, but you can always reach out to me through my email.
All right, sounds good. Thank
Thank
you.
you.
Okay, next up on the agenda is a presentation on the Cell by Gene resource. And giving that presentation will be Maximilian Lombardo. Maximilian, where are you?
Michelle, if he's not here, I can also just run through some of their profiles as well.
Okay. I don't see him online, so maybe something detained him. So yes, that'd be great, Brian, if you could do that.
Okay. Yeah, just give me a minute. I'll get off this other call. Sorry.
Okay.
Okay. Sorry, folks. I hope you don't mind the audible on this one. All right. So yes, I think the very important point that as the BICCN, obviously we've noticed the fact that they generated a massive amount of data. The cool thing is that now there's all these tools that are popping up. So we saw Brainome, Catalyst, very important sites for the epigenetic data. The other tools that explore the single cell, the transcriptomic data in terms of taking a look at UMAPs, the single cell portal, I encourage you to take a look at that. That's something associated with the Broad Institute. Another initiative coming from the Chang Zuckerberg initiative is the Cell by Gene. So let me, I have a listing of various profiles that they built for the BICCN thus far. One moment, please. All right, sorry, I don't know, Michelle, if you had other just general announcements while I'm looking up this information, but.
Sorry, I was muted. I will take this opportunity to say that that we will be sending out an evaluation form that it's an online SurveyMonkey survey to evaluate the workshop. We'd really appreciate you filling that out. And so it's just a reminder of that. I mentioned it earlier. We will be sending it out right at the end of the workshop, and we'll also put the link here in the chat. If anyone has any questions on anything so far.
Okay.
All right.
All right. So yeah, as I mentioned, for a couple of the papers that have come out, we've minted data set identifiers.
Brian,
oh, I'm sorry. Am I not sure yet?
No, I hear you. Maximilian is just now joining.
Fabulous. Perfect.
Yeah, he's here now. Hello, Maximilian. Perfect timing. We were just starting your part. We're running a few minutes early today.
Okay, got you. I hope I wasn't holding up anything.
Nope, you got here just in time.
Perfect. Should I go ahead and share my screen?
Yep, absolutely.
Okay, give me 1 second. Move Zoom out of the way and okay, let me see how to do this. Cool. Do you see a big red slide on your
Yes.
computer?
Yep, looks good.
Cool. That sounds great. Well, thank you for having me. It's super nice to be here giving this presentation. And before I get started, I'm just gonna deposit some, if I can find the Zoom chat, which it's evading me right now. Let me see if I stop the share. I'm just gonna deposit some relevant links and contact information. So like, if you have any questions after the talk, please feel free to, you know, ask me questions about cell by gene. My name is Max. I'm a product application scientist within the cell by gene team. And so I'm really responsible for interfacing between the community of scientists who are using cell by gene, relaying messages back to the engineering team and product team. And yeah, just hopefully helping people learn how to use cell by gene and in turn making it a better product. And so, you know, basically the kind of intro that I'm going to give today is an introduction to cell by gene. And then I'll introduce also a couple of different use cases that we've been evolving the cell by gene product suite towards. So the first would be kind of this collaborative data exploration mode. So I'm definitely in the right place at the BICCN Data Exploration Workshop. And then we also have this other use case where we've been developing this publishing and data hosting platform. And so, you know, before I actually introduce cell by gene, I want to talk about the development principles. So we aim to develop a product that is basically driven by interests in the biological community. So we want obviously whatever we do develop to be appreciated and wanted and needed by the biological community. And then in turn, when we do try to decide like how, you know, what direction the product is going in, we are cognizant of the fact that the field is changing very rapidly right now. And so to that extent, we've tried to build cell by gene to be a little bit future proof in that basically the tool is somewhat analysis and data agnostic. So especially talking about single cell data, you can actually take in you know, anything that is in this kind of like tabular format. And then we also have done a lot of work to make sure that it works at different scales. And so, you know, I think it's one of the few tools out there that can really provide a smooth user experience for some of those larger, like million cell, single cell data sets. And I guess just because I don't know the background of everybody on this talk. So cell by gene is basically a tool that is specialized for visualizing single cell data. And so we'll get into this a little bit. So since it is specialized for visualizing single cell data, I thought it would be useful to take a look at how that data is typically organized, especially within the major computational tool chains that have been developed to perform various analyses on those types of data. And so in general, what you get is like, you know, three kind of like main matrices. So you'll get the data itself, right? So this will be like a cell by gene count matrix or a normalized count matrix. So there are various basically results from the experiment. In addition to that data matrix, then you'll also get cell metadata. So these are annotations of individual cells that have been collected in the data set. These might be tissue of origin. They might be after you've kind of analyzed the data set, you might find that there are different cell types. So you can put kind of like clusters or like cell type IDs on individual cells. And there are, you know, a bunch of different types of cell metadata. Here we're also calling like the embeddings that, you know, are the typical TSNE or UMAP visualizations, also referring those as kind of like cell metadata features. Additionally, you might have feature metadata. And so these are basically just gene names, but then we're also a little bit more well, perhaps I shouldn't use the word boring, maybe less exciting, is that we're, standardizing those gene names and their links to ensemble IDs. So those can be there. I think that people can also basically calculate different metrics around individual genes. So those can be useful too. But this is what the single cell communities and single cell tool chains have kind of represented on, landed on this type of representation for single cell data. And then to some extent, we try to represent that in the cell by gene user interface. So here we get metadata over on the left-hand sidebar. Particularly, we get like categorical observations. So like the tissues that I was talking to before, or maybe the assays that the cell was collected from, or the cluster label, for instance. This is a BICCN data set, by the way. This is the from the Mukumel lab, the integrated transcriptomic atlas of the primary motor cortex. So in addition to the categorical metadata, which take on discrete values, you can also have numerical metadata, right? So they might be different QC metrics related to cell health, or related to like the sampling that was done during the experiment. So here we have like cell size. Additionally, in the center pane, we have kind of where all the action is going down. So here's the embedding, which is, the kind of typical representation when you're looking at a single cell atlas. And then on the right-hand side, we have basically gene search and gene sets. And I'll introduce those in a demo a little bit later in the talk. But this is kind of, I will say one more thing, this is kind of how you're interacting with your data matrix from the slide that I showed you before. And just a brief example. So it's usually pretty interesting to be able to view the relation of these categorical metadata in relation to the embedding that's been computed, right? So here I'm just coloring by cell type. We see that basically there's a bunch of different collections of cells or clusters of cells that have all been annotated as the same cell type, and they all kind of occupy the same space on the embedding plot. And we'll see a little bit more of that in a little bit. Getting a little bit deeper into what the different buttons and tools do in the cell by gene interface. So here on the left-hand side in these metadata fields, so here I've expanded the BICCN cluster label, so just the cluster. And then we see that we get a list of all the clusters that have been annotated in this data set. To the right of that, you see the color that corresponds to that on the embedding. And then you also get a cell count. So that's pretty useful. If you utilize these checkboxes, you can select or unselect those cells, which is useful for creating different subsets of your data set. We've covered coloring by variable, but you'll see these little droplets kind of all over the place. And so whenever you see those, you can color by that particular attribute. We have a differential expression toolbar. And so here is where you would set the first population and the second population. So essentially kind of like a numerator and denominator here. And here's where you would actually calculate that differential expression after you've assigned those populations. Here's a button that we use to create subsets of the data sets after you've made a selection of cells, either based on a categorical or numerical attribute. And here is a button which allows you to reset to the whole data set, which I'll demo all this for you shortly. Here we get lasso selection. So it's essentially selecting cells just by using your cursor to draw a little loop around them. Here you get a zoom and pan tool. Here is to display categorical names over the cluster centroids. Here's a little clipping tool, which I think I'll just explain in the demo. And then here, just forward and backward buttons. So in case you want to undo or redo actions that you've taken, it allows for that. And then finally here on the right side, as I've already kind of mentioned, here's where we look at different genes and gene sets. I inadvertently explained this already, so I think we can get on to the next slide. And so one thing that I'll note is that there are kind of, well, let's say, two different versions of the Cell by Gene Explorer. So this version that I've shown you, this is accessible via our publishing platform, which I'm kind of talking about later in the talk. But so this is basically a link that you can access online. You don't have to do any setup or anything like that. There's another version of Cell by Gene, which is kind of the original version of Cell by Gene Explorer, which is, we call it the Desktop Explorer. So this requires that you have to kind of download or pip install the program and then work with the data set locally. And so when you do that, the one reason you might want to do that is because it offers this additional functionality where you can essentially create custom annotations based on custom selections of cells that you've made in the Cell by Gene Explorer. And so to access that functionality, you would go to create new category, and there are a couple other steps, and I'll demo those briefly. But with that, let me switch briefly to another tab. And so here we are. I hope this screen looks familiar. So I've kind of, already explained the layout a little bit. So what we can go ahead and do is just color by, see how we do some of these actions that I was talking about before. So we can color by the BICCN cluster label. We do have kind of the color coding, although that can be a little bit, difficult to see. The nice thing is that when you basically scroll around or move your cursor over the names of these different cell type labels, then the corresponding cells on the embedding plot do get highlighted. So that's cool. If that's not good enough for you, there also is this button. So basically, when you click this, it probably looks a little nicer when there's less cell types present, but it essentially displays those cluster labels over the centroids of the cluster, or does its best guess at the centroid of the cluster. Sometimes clusters are kind of split apart, so you end up with a label in between the two populations, but it usually works out pretty well. I'll go ahead and turn that off. And that button, I believe, is only active when a categorical attribute is, when you're coloring by a categorical attribute. Let me see. Oh, and then beyond this, so we can also, as I was mentioning before, we can use these checkboxes to kind of select and deselect cells. So if, for instance, we wanted to deselect all of the cells in the pane, so notice that the intensity of the points, or like maybe the weight of the points, got minimized or diminished once I deselected all the cells. And so I can go and maybe select just like a really, a population that's interesting to me. So maybe I just want to take a look at astrocytes, and then doing that selection, I can finalize that by using the subset button. And now we've got just the astrocytes here. So that's pretty useful. It's pretty useful in creating particular selections of the cells that allow you to focus the analysis a bit more. Note that I just used the whole pie little icon here to bring all the cells back. We can also check out the kind of interaction between different categorical metadata. And so one thing I'll do, I'll switch over to the cell type, which is this one has been annotated or translated the CZI schema, so it's a bit more coarse than the BICCN cluster labels. But what we do see here is the major cell types. And so note that I've colored by cell type. And then what I'll do is I'll actually open up another categorical attribute. And so what we can notice now is that we get this extra bar here. And I've opened up assay. And so what this essentially is it shows for each assay type, what is the distribution of cell types that are present there. And so I thought that the kind of cool thing about this particular comparison, or this particular scenario, was that oligodendrocytes and some of the other accompanying cell types beyond neurons seem to be most represented in this 10x3' V3 sequencing. So wasn't sure, perhaps it's due to the fact of the number of cells that were collected. I'm not totally sure on the experimental design of this particular study, but I thought, you can start to interrogate those types of questions like, oh, is my cell type present under this particular condition, right? So you might imagine a scenario where you have a disease state versus a normal state, right? And then you can see, oh, there's actually depletion of the cell type in the disease state. Um, moving on from that, we can also start to take a look at, let me close these real quick. We can also start to take a look at the cell numerical metadata. So here I'm looking at cell size. So it's variable, but not unexpected. And we can, do the same thing again. So I just opened up assay while coloring by the cell size. So this time we get a bit of a different view. So this is a continuous attribute. So we're seeing a histogram instead of this bar chart, but we do get to see this these values, the size values partitioned over each of the assay conditions, and we can see that some of these outliers here, some of the high size cells are only for the are only present in the 10x either V2 or V3 sequencing, which makes sense. I think that the SmartSeq is not collecting or not detecting as many molecules as the 10x for the 10x's. So that's cool. I think it makes sense. Hopefully it gives you guys some examples of how to, start to look at how your data is varying across experimental conditions or assays. With that, I'll switch over to looking at features. And so what we can do here is I'll just uncolor by this. So here's the gene search bar. So you basically start searching in your gene name. The cool thing is that it auto suggests based on the prefix that you've already entered. So like here, I'm looking for somatostatin. But the kind of cool thing is that, the receptors for somatostatin are coming up here. Obviously, it only works when the when the peptide and when the peptide and the receptor have the same prefix, but it can be nice. It's also nice, I think, when you're looking at different members of a family. So that's a feature that I found useful. But we'll go ahead and we'll take a look. And so I've just entered somatostatin, or I've made the selection. So we'll get this little row in the gene list that pops up. We can go ahead and expand that. So it gives us a little bit fuller of a view. You'll see that what's been brought up is a histogram of somatostatin expression across the whole data set. A lot of these will look like they have, huge numbers of zeros. So I think it's just you can't really avoid it in single cell data, especially when when your population that's expressing the gene is a small subset of the total data set. But we can go ahead and color by this gene. So we're seeing that it's really a great marker for this particular, for these few cell subtypes in the embedding plot. I would say that it doesn't look as good as it could be, just because there are a few outliers here that are basically throwing off the color scale. So what we can do there is use this clip tool. We can go ahead and just clip out the values that are in the 99th percentile or above. And so there we get, it's obviously a similar pattern to before, but then we get just I think a nicer picture. This one's publication ready. What I do also want to do is that, we can make selections of cells based on these expression values, right? So we can do something like this. I call this brushing the histogram. So I brush from a range of 3 to 4 here. And then we can go ahead and use that subset button to finalize that selection. And so here, obviously there's a bit of noise here, but we've really, narrowed down our selection to these somatostatin positive cells that were, really just occupying the small part of the embedding. So it's a pretty useful feature to be able to select based on those expression values. I'll undo that briefly. I think the only thing I want to note is that we can again, and I hope there's not too many labels here because that can sometimes take a while to compute, but that we can basically get access to these histograms and look at this gene expression now by partitioned by different, basically partitioned by whatever categorical metadata you choose to open. So in this instance, I've opened the BICCN cluster label, and we're not seeing a lot of expression here until we get, sorry, S is way far down the alphabet. So p-valbumin, but really in the somatostatin. So basically all these various subtypes that are expressing somatostatin, or which have been annotated as expressing somatostatin. So here, this is kind of a trivial example, but it is useful to be able to like look at the expression of your favorite gene in relation to the annotations that you've made on this data set or that have been previously made by other authors. I'll go ahead and close that up. And oh, so the next feature I do want to introduce, I'll uncolor by this. So the next feature I want to introduce, actually, yeah, let's keep this in somatostatin cells. Let's say we're interested in what is, because we do have these, this whole collection of clusters that's expressing somatostatin. So let's say we start to get interested in what is driving, you know, like these cells to group together, for instance, and like these cells to group together, you know, on their own, you know, and separate from the rest of the somatostatin populations. And so what we can use to do that is like start to run differential expression tests on various selections of these cells. So what I'll do is I'll highlight this cluster, or I'll lasso select this cluster. I'll assign that to population 1. I'll do the same thing for this one right here and assign that to population 2. And let's see, I actually think I made the wrong selection. I'll select a little bit more and assign that to population 2. And let's go ahead and run a differential expression and see what happens here and what pops up. It'll take a little bit because this is a rather large data set. I think it's like in the neighborhood of like 500,000 cells. And while that's computing, are there any questions before, you know, before I go on? Cool. All right. Let me try that one more time. Cool. And so what we've seen here is that after we've run the differential expression using our kind of custom selections of different populations, we get now these 2 entries which appear in the gene sets menu. And so each one of these corresponds to genes that are highly expressed in population 1 and then population 2. We can expand each of these so that now we get not only the histogram for the, not only do we get, well, this is still loading, but we'll get a histogram for the gene set itself in total. And then we get also basically detailed views of each of the individual genes. And so I'll go ahead and expand the second one as well. So we'll kind of wait for these guys to start loading. But here, so here is the basically histogram of, I would say, gene set scores for the population 1 high gene set. And so essentially, I believe how this is calculated is just an average of expression across the different genes that are in this gene set. And so the cool thing is that we can start to color essentially by this gene set. So here's the population. Let me go again. And so here, I think we have the population 1 high, and let me color by population 2 high real quick. So essentially we do have the coloring of this gene set, and we see that there's distinction between the first population that we chose and the second population that we chose. Here we're coloring at the gene set level. We can also color by individual genes in there. And so here's a little bit more what we're expecting, where we see basically an individual gene. That gene's expression level is quite high in the first population that we chose. We can look at another population in the second gene set, which is population 2 high, and see that between those 2 populations, this has the much higher expression. So that's pretty much what we expect. We can do some other interesting things if we want to like interrogate the relationship between these 2 populations a little bit more. So we can go ahead and take a subset of those 2 guys And then we can do some interesting bivariate plotting. So if you saw these X and Y icons here, they essentially allow you to construct a FACS-like bivariate plot. So here I'll set man1A to the x-axis and gal NTL6 to the y-axis. And so this is kind of just another view that shows us that the expression of these two markers is, well, there's no co-expression, right? We don't see a lot of cells that are have both high levels of gal NTL6 and man1A. The other cool thing about that is that we can color by each of those genes. So basically the coloring on there works. And and we can also, if we wanted to start to perform selection of those cells. So for instance, if we wanted to like take man1A positive and gal NTL6 negative cells, we can just go ahead and make that selection on our histogram here. So here I've selected basically all the positive cells for man1A, and then I'll go ahead and do the same thing, although the selection is already kind of implied, but I'll just select the 0 population for gal NTL6. And then essentially after we've made that selection, now we just end up with those man1A positive cells. And yeah, so that's kind of a, a cool function that you kind of start to drive down deeper into like, what are the transcriptional differences between two distinct cell populations? And then how do I, get down to just those subsets that I'm interested in. With that, I think there's just one more piece of functionality, or maybe two. So I'll go ahead and remove this. I'll bring back the entire data set. And so we've already been kind of introduced to the concept of gene sets here via the calculation of differential expression. And let me say before I move on that we can edit these names so you're not stuck with the POP1 high and POP2 high. It could be, interesting somatostatin population 1, or, you can basically edit the annotation there or edit the gene set name and also description. I'll go ahead and delete these and I will create basically under this gene sets toolbar, there's also, this gene sets kind of row or section, there's this create new. And so here I can create a new, just totally custom gene set. So I don't need to run differential expression to get a gene set. The particular list of genes that I've chosen are supposed to be good markers for oligodendrocytes. So if I can spell today, then I'll go ahead. Well, I'll just skip the description. And then I'll add in these guys. So this is a comma separated list of genes, which hopefully are good markers for oligodendrocytes. We can go ahead and expand that. It takes a little bit to load. So we'll wait a little bit. But here we are. And so let's color by the gene set. So that's looking pretty good. Here's our oligodendrocyte population. Again, we can color by different, individual genes in the gene set and see where they are. And so there's actually some interesting patterns here. I believe these towards the end here are like precursor oligodendrocyte precursor cells. And I think over here are more mature oligodendrocytes. So that's a pretty interesting pattern to observe, and especially when you dig down into the individual genes there. And then the other thing I do want to highlight is that even at the gene set level, we can go ahead and we can check out the distribution of those gene set scores across any categorical metadata type. So for instance, I've just opened up cell type. These are kind of the core cell type annotations. And we see that, astrocytes not there, endothelial cells not there. But yeah, lo and behold, we do see a good, right shifted distribution in the oligodendrocytes and the oligodendrocyte precursor cells. That is that is how do I phrase this? That's most of the things I would say that are available in the hosted Explorer. I think the one thing that I didn't cover that isn't particularly relevant to this data set is that you can change the embedding choice. So, if you have like UMAP, TSNE, PCA, or different pseudotime embeddings, you can, basically select all of those here. I'll switch briefly to an instance of the desktop application just to highlight the fact that we do have this extra button. And so here we would create a new category. We'll say Max's annotations. And so we could assign cells from the different metadata categories there. And so I'll go ahead and try. This one I didn't prepare as much, so I think someone may need to ask me questions about this afterwards. And I don't use this feature that much, so I'll have to I guess the one thing to note is that there is this create new category thing here. I'll forward you to some documentation because that was not a very strong show of the feature. With that, I'm going to switch back over to the presentation and just run through really quickly. Basically, so I've kind of introduced these versions of the application, and I think the thing to note that I've kind of already alluded to is that there are essentially these different use cases and then different deployment modes. So for that annotation function, which was the last thing that you didn't quite get to see, you can run it locally. So this requires that you both install it and run it from the command line, also that your data set is hosted locally. So obviously there's compute restrictions there. There are also options to set up that annotation functionality or the Cell by Gene Desktop Explorer in a self-hosted environment. So you're basically, setting up your own hosting infrastructure. We do have recipes for that, although we don't explicitly support or provide user support for that use case. And then finally, what I'll get a little bit more into is our publishing use case, where CZI essentially hosts single cell data sets. So they could be transcriptomic, epigenomic, spatial. We host those. They're typically associated with publications, and we also pay for the compute for hosting. Beyond that, we also guarantee that the links that are generated behind those visualizations are permanent. So you can use those, for instance, in your publication. And I guess I'll skip over the history portion of it, and I'll just show you actually what. So I'll show you what the portal looks like. So if you go to that link that I deposited right in the beginning of the talk, so cellbygene.cziscience.com, if you go there, you'll be greeted by a page of collections. Each of these collections represents a publication typically. And you'll see with each publication, you'll see various data set and collection level metadata. So these could be the tissues that are represented, the assays that have been used to profile cells, disease states represented, organisms, and then cell count. If you click into a collection, and here I'm going to the Mucomel collection, going to the Mucomel collection, and we can see that now we have all of the data sets associated with that publication and that collection. So in this instance, I believe we see a lot of data sets because we essentially have the integrated atlas and then a few data sets which correspond to the different assays that were used, I believe. Similar metadata is available here. And then the thing to note is that you have two new icons. So here is a download button, this cloud with a download arrow in there. And then here is a Cell by Gene Explorer icon. So essentially, if you click on that Cell by Gene Explorer icon, that brings you to the visualization that we're just at. And I can demo that briefly after this next slide. So here, this is what the download dialog looks like. And so it's pretty cool. So it allows you to download into the popular formats for analyzing single cell data. So ANN data and Seurat. So we used to have Loom. It didn't seem to be that widely used. So we've recently gotten rid of it. Like, curious to hear what, if there's any Loom folks out there. And then additionally, what we have is also curl commands. So if you are comfortable with the command line, we do provide the curl commands to allow you to download the set that way. every data set is available as a visualization. And so let me just go back there and demonstrate. So here we're on the site actually, and we're in this integrated transcriptomic and epigenomic collection. And so we can go ahead and click on any of these. And so maybe we'll do a new one just to check out what it looks like. But if you click on that, it takes a little bit to load depending on the data set size, but yeah, we basically get the visualization, no programming required, no setup required, no data download required. So it's really like a very nice way to quickly be able to like look at data sets, check out what's interesting, maybe compare your data set with something that's already been published. And then obviously we're supporting computational reuse cases, right, through that download feature. So we make it really, really easy to essentially download data sets and then put them into your tool chain of choice or your favorite computational biologist tool chain of choice. With that, I really want to thank all of you for taking the time to listen to me. I hope I didn't go too far over, but I hope you enjoyed it. And I do want to give a shout out to the team right now. So obviously all of this work or this awesome product would not be available without the hard work of all of these people. So I want to give them a shout out. And yeah, I'd like to open up to any questions that anybody may have. And thanks again.
Thank you, Max. That was great. We did get one question in the chat while you were speaking. It's, does Cell by Gene support Lumpy formats?
Oh yeah, so it doesn't. So essentially what you would have to do is if you have a LumPy object already, then what you would need to do is just convert it to ANN data. And it doesn't, so for instance, like Cell by Gene Desktop doesn't support Seurat objects natively. So you'll always have to go to this conversion to ANN data. And then once you've got to ANN data, then that's an object that Cell by Gene will recognize.
Okay, great. Any other questions, anyone? It's been a quiet group today. I haven't had too many questions.
That's cool. I mean, I did a lot of talking, so I did it for the whole group. I do want to just mention that, like, yeah, I'm always happy to like receive questions over email. So like if something comes up later, or you're like, Max, I really want to know about annotations workflow, that was rocky in there. Yeah, feel free to reach out for me for that. And yeah, so I'm super excited to have some new users in Cell by Gene, and yeah, I enjoyed today.
Great, thank you. All right, well, folks, that brings us to the end of the workshop. I am going to paste into the chat right now the link to the survey. And everyone, please, it would be ideal if you would actually go to the survey right now and fill it out while it's fresh in your mind. It's very short, just 5 questions. Most of them are just radio buttons. Actually, you could choose to make all of them radio buttons if you didn't fill in any comments, but thank you if you would please do that as soon as you can. You will also be receiving an email from Joe, who sent out the workshop information a couple of days ago. You'll be receiving an email from Joe with the evaluation survey. Next week, once the recordings from today get processed, we will post those to the website, and we will send an email when that's done, and we will also send along all of the other links that the presenters shared with their information, so you have all of that. I want to thank all the presenters again. I really appreciate you taking the time. Thank you, attendees, for coming. Does anyone have any final questions? All right, thanks again. And we will, I think, see many of you attendees again next week at the workshop that is next Thursday. So thanks, everyone. Bye-bye.
Thank you. Thank you, Michelle.